السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. الحمد لله رب العالمين، حمد الشاكرين حمدا نعمه ويكافي مزيدا. يا ربنا لك الحمد كما ينبغي لجلالك وعظيم سلطانك. اللهم صل على سيدنا محمد في الأولين، اللهم صل على سيدنا محمد في الآخرين، اللهم صل على سيدنا محمد في النبيين، اللهم صل على سيدنا محمد في المرسلين، اللهم صل على سيدنا محمد في الملأ الأعلى إلى يوم الدين. اللهم صل على سيدنا محمد الفاتح لما أغلق والخاتم لما سبق ناصر الحق بالحق والهادي إلى صراطك المستقيم وعلى آله حق قدر ومقدار العظيم. سبحانك اللهم ربنا هب لنا من أزواجنا وذرياتنا وتلاميذنا من أهل العلم وأهل الخير، ولا تجعلنا وإياهم من أهل الشر وأهل الضير. Ya Allah, ya Rahman, ya Rahim. Hari ini kami berkumpul di acara International Conference on Life Science and Technology 2022, Departemen Biologi Fakultas Matematika dan Ilmu Pengetahuan Universitas Negeri Malang. Sepenuhnya kami laksanakan, ya Allah, dalam rangka mensyukuri nikmat Agung-Mu pada kami. Kami berharap dengan kasih sayang-Mu melalui ilmu pengetahuan, hikmah, dan akal yang Engkau berikan. Oleh karena itu, ya Allah, berikanlah kepada kami petunjuk untuk menjalankan perintah-Mu dan menjauhi segala larangan-Mu. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. وادخلنا الجنة مع الأبرار يا عزيز يا غفار يا رب العالمين. وصلى الله على سيدنا محمد وعلى آل سيدنا محمد. سبحان ربك رب العزة عما يصفون، سلام على المرسلين والحمد لله رب العالمين. بأسرار الفاتحة والله موفق إلى قوم الطريق. ثم السلام عليكم ورحمة الله وبركاته.
Ladies and gentlemen, next agenda, we are going to listen to the event report that will be delivered by the head of Biology Department, Universitas Negeri Malang, Chairperson of ICOLIS 2022, Professor Dr. Sri Rahayu Lestari, MSI. For Professor Sri Rahayu, the time is yours.
Welcome to our conference, the 5th International Conference on Life Sciences and Technology ICOLIS, which will be held 2 days from today and tomorrow, August 30 to 31, 2022. It gave me immense warmth and great pleasure to grace all of your presence in the interest of the entire committee. It gave me tremendous commitment to be presenting the welcome speech among the most esteemed personalities who have won appreciation in their respective fields. Before we begin international conferences, I would like to express my heartfelt gratitude to all of you sincerely committed to this event to make it a success. This event would have been impossible without the support of each and everyone present here. This is our first hybrid conference organized by Department of Biology, Faculty Mathematics and Natural Sciences, Universitas Negeri Malang, Indonesia, sponsored by Nikon and GeneCraft Labs. We are also supported by co-organizers for many national and international universities, which I cannot mention one by one. Ladies and gentlemen, in this special occasion, I also would like to thank all distinguished keynote speakers who come from 12 countries. Dr. Prasetya Papankaran from University Technology Malaysia, Malaysia. Professor Dr. Champan Kanchau from Chulalongkorn University, Thailand. Dr. Muhammad Aziz, the University of Tokyo, Japan. Dr. Mario Rosario Guarcino, National Research Council of Italia, Italy. Professor Dr. Rob Harris from University of Liverpool, United Kingdom. Dr. Reti Ratnawati, Indonesian Physiological Society and the National Commission of Violence Against Women Indonesia. Dr. Yi Wan Lin from National Tsinghua University, Dr. Mulyono Pangestu from Monash University, Australia. Dr. Asuncion G. Beltran from Tarlac Agricultural University, Philippines. Dr. Deka Sembitiawong from Prince of Songkla University, Thailand. Professor Corny Peters from Utrecht University, Netherlands. And Dr. Hendra Susanto from Universitas Negeri Malang, Indonesia. Over 180 participants, ranging from students, lecturers, researchers, on life sciences register in online or offline mode. Hopefully, ICOLIS 2022 will enhance our insight and contribute in addressing global of life sciences challenges. Therefore, we proudly present this annual conference under the Building Network of Sustainable Research to Promote Global Conservation and Responsible Use of Natural Resources. The fifth ICOLIS 2022 is successful because the support from many parties, including the Rector of Universitas Negeri Malang, the Dean of Mathematics and Natural Sciences, Universitas Negeri Malang, lecturer in Department of Biology, and all dedicated committee who have been doing fantastic job to make this event run smoothly. Finally, I would like to convey sincere welcome at Universitas Negeri Malang. Have a nice and insightful today's conference to all of you. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for Professor Sri Rahayu. Entering the next agenda, addressing. The address is going to be delivered by the Vice Rector 1 of Universitas Negeri Malang and continue for the opening of the conference. For Professor Dr. Haji Budi Eko Sujipto, MED, MSI, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A very good morning, ladies and gentlemen, honorable speakers, and our audience joining us online from around the world. I am pleased to welcome you to the 5th International Conference on Life Science and Technology 2022 in a hybrid format. In this year conference, there are 12 keynote speakers from whom I cannot mention the name one by one. They come from 10 different countries, namely Indonesia, Malaysia, Thailand, Netherlands, Philippines, Italy, Japan, UK, Taiwan, Australia, Spain. I hope from our keynote speaker, we can get not only various perspectives on the progress of life science and technology in other countries, but also strengthen our network in contributing ideas and even designing collaborative projects in solving world problems. Ladies and gentlemen, the theme for this year's conference is network of sustainable research to promote global conservation and responsible use of natural resources. We have been building a network, one of which is through this annual conference. However, only few of us are aware that this has become an urgency, one of which is listed in Sustainable Development Goals number 17, Partnership for the Goals. So that with this conference, I hope that all participants, researchers, educators, and policymakers can inform and inspire other participants in solving problems that are currently. To support this program, Universitas Negeri Malang will open wide collaboration either with national or international institutions to jointly create useful contribution to the work of research, academics, and society by opening several funding channels. Therefore, we always open for invitation of an impactful collaboration. Distinguished guests, ladies and gentlemen, the fifth ICO list 2022 is successful due to the sum of many important parties who have contributed in planning on and organizing the conference very well. I would like to take the chance to extend my greatest appreciation to all the committee. I also appreciate and thanks all the participants for attending our conference, either who attend this venue or join us by Zoom. Enjoy the fifth ICOLIS 2020. I hope that you find it fruitful and exciting. By saying Bismillahirrahmanirrahim, I'm pleased to open the fifth ICOLIS 22. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi
wabarakatuh. Thank you very much, Professor Budi, for delivering the address.
Ladies and gentlemen, before we step to the main agenda, allow us to thank our sponsor who have supported this conference. To the operator, please show the sponsorship video.
Ladies and gentlemen, the next agenda, let's take a photo together first. All the participants in the Zoom meeting, please turn on your camera. Thank you.
And for the documentation team, the time is yours. All right, for participants in the Zoom, I'll guide you first to take photos. Please turn on your camera first and take a good position. All right. For the slide one, I will count 1, 2, 3. All right. Okay, for next slide. All right, thank you for everyone who has opened your camera. Now, let's take photos together here. For the documentation team, the time is yours. All right, thank you. For ladies and gentlemen, now we step to the main agenda, which is the first plenary session. In this plenary, we have Professor Dr. CMG Corny from Utrecht University and Dr. Ma Asuncion G. Beltran from Tarlac Agricultural University as the keynote speakers. And also, Mr. Fauzi Akbar Anugrah, SSI, MSI, as the moderator in the session. For Mr. Fauzi Akbar, the time is yours.
Okay. Good morning, ladies and gentlemen. Um, yeah, it's nice to see you all here. So yeah, today we're going to start. We'll kick our event for the plenary session, the first plenary session. So in this session, we will have two speakers. One in this venue directly, and then one is from online. So yeah, I will try to introduce first our speakers. For the first speakers, I remember back in 2030, I already told this to him in person, but I will try to share with you all. So back in 2030, when I'm in undergraduate, I'm I really interest. I have an interest with microbial and plant interaction. So I'm trying to find any resources to get my knowledge related with the plant and microbe interaction. I found one of the papers that written by who is the person? The person is Professor Corny Peters from the Netherlands. So I am a big fan of him since very long time ago. And can you imagine what I'm feeling right now? I'm meeting him in person. So yeah, this is my honor and also my privilege to represent you about him. And Corey Peters is a professor of plant microbe interactions and scientific director of the Institute of Environmental Biology of the Faculty of Science, Utrecht University, the Netherlands. His research group investigates how the plant immune system protect plants against microbial pathogens and insect herbivores, and how beneficial microbes in the plant root microbiome stimulate plant growth and health. Current research is focused on plant beneficial functions that are encoded by the root microbiome, the role of plant genes and metabolites like coumarin that aid in the plant defense hormones. With his research, he aims to contribute to grand societal challenges such as food security and sustainable agriculture. So, Corny Peters studied plant breeding and plant molecular biology at Wageningen University, where he graduated cum laude in 1988. He performed his PhD research in Wageningen on the molecular basis of pathogenicity on the potato lead blight pathogen Phytophthora infestans, which is I also doing this. I'm used Phytophthora infestans in my first ever research. And then after obtaining his degree in 1993, he moved to Utrecht University first as a postdoc fellow and between 1998 to 2004 as an assistant professor in molecular phytopathology at the Department of. In 2004, he was appointed as full professor of plant microbe interaction. In 2010, he was awarded an ERC Advanced Investigation grant by European Research Council. His research group pointed research on unraveling the rhizobacteria-induced systemic resistance signaling pathway and the role of phytohormone crosstalk in the regulation of plant immunity. In 2013, he was elected as a member of the Royal Netherlands Academy of Arts and Science. Since 2014, he is an ISI highly cited researcher. Besides heading the plant microbe interaction group at the Department of Biology, Corny Peters is scientific director of the Institute of Environmental Biology of the Faculty of Science at Utrecht University. Nationally and internationally, he fosters plant microbiome research. So give big applause to him, and I invite you here.
So there's no laptop here, so I cannot the slides. But I first would like to thank for the very nice introduction. I didn't realize that I had such an influence on the life of people here in Indonesia. So it's really nice to hear that I inspired you at a young age. So that's really nice to hear. Yeah, I'm from Utrecht University in the Netherlands, which is in the center of the Netherlands. And yeah, it's nice to visit a university which has very high on the agenda the teaching of the young generation. I'm also a teacher in biology. And well, also at Utrecht University, teaching is very important. We do a lot of research, but teaching the young generation is also very important. And that's why I found it also very nice that a delegation from this university came to visit Utrecht a couple of years ago. And we could change ideas about how to do teaching in biology and how to do science. So that is really nice. Yeah, so this is the first slide. And I would like to point to certain things in the slide. So it would be handy if I would have a laptop here. Is that possible or not? Six people are trying to make it work. Okay. Yeah. Yeah. I was just saying next slide. No problem. Okay, we are going to do it like this. Every time I want to make a change in the slide, and I say next. Okay. Yeah. Okay. So what you see here is me, but if you go to the first slide. Sorry for creating such, but sure they'll be fine. It's still early. Oh yeah, that's right. And you know, it's very nice that we can all be together again and meet each other instead of sitting at home behind a computer. So it's really nice to be live present again. And then of course you need you have to get used of doing things live again. So that's why maybe the startup is a little bit okay. Well, what you see here is basically the city of Utrecht, University of Utrecht. We have a very nice tower. Top is lost. I don't know where it is. The tower was already built in 1290, so that's 700 years ago. So it's a very old tower. We have a very old city, and well, the city center is really nice. So if you ever have the chance to visit the Netherlands, please come to Utrecht because it's a very beautiful old city. So next slide, please.
Yeah, so I already was welcomed yesterday by people from this university. We had a very nice dinner and we visited Toko Un. And actually, if you look on the left bottom, that is my daughter 10 years ago. We already visited Malang 10 years ago. And in those days, she was in the front of Toko Un here in Malang. And we visited it again yesterday, so that was really nice. So, well, Malang is a really beautiful city, so you can really be happy that you are here. The next slide, please. Yeah, I would like to talk. I don't know what happened with actually, but is it possible to get rid of it? No, well, anyway. So what we study, and which is not mentioned because there's a black thing over it, we study the root microbiome of plants. So why is the microbiome of plants very important? Well, we all know that plants photosynthesize. Next, please. And when you get photosynthesis, plants use a lot of photosynthase, a lot of sugars. Next. And those sugars, they are exported also downwards to the root system. And there they are excreted into the soil, into the rhizosphere, as we call it. And in the rhizosphere, these root exudates are lost, you would say. You would say it's very stupid that a plant secretes all the sugars that it makes into the rhizosphere. But they do that because they want to feed the microbiology that is in the soil and that is selected to the root system. Because many of those bacteria and fungi that are around the roots are able to mobilize nutrients in the soil, and they can provide food for the plant so that the plant can grow better. If you take 1 teaspoon soil, then typically there is 1 billion of microbes in that 1 teaspoon of soil. So there are many, many microbes in the soil, and the plant roots select those microbes to their roots. Many of them are beneficial, so they provide benefits to the plants, so that the plants, next please, so that they can grow better because they take up more nutrients, but also these microbes can stimulate the immune system of the plant, and they can provide protection against, for instance, drought stress or other types of stress. Next, and in Utrecht, okay, now I can use my chance. Interrupt you. Yeah, let me see. Okay, yeah, I need my glasses now because I'm old. Okay, you see the pointer? Yeah, okay. So what we do in Utrecht, we study the immune system of the plant. And let me see whether I can do this correctly. Next. Yeah, so a very important phenomena that we study in Utrecht is the fact that when certain microbes colonize the roots of the plant, then suddenly the immune system of the plant becomes much stronger so that it can protect itself against all kinds of pathogens, also pathogens that live on the roots, on the leaves. And we call that induced systemic resistance, or ISR. That is a phenomenon that was discovered in Utrecht. And that is the phenomena that when certain microbes are colonizing the roots of the plant, the whole plant body becomes better protected against all kinds of pathogens and insects. Well, we found it extremely interesting and important, because if we can make use of those beneficial microbes that live on the roots, then we might be able to stimulate the immune system of the plant and provide more resistance against different types of pathogens without using chemical fungicides, chemical pesticides. So we can reduce the use of chemical antimicrobials, so that in agriculture we can make it a little bit more sustainable. Yeah, so that's where my talk is about. So we do not work a lot on crops, but we would like to learn from nature, because this is a phenomenon that exists in nature for all plants. So we study the model system Arabidopsis thaliana, which is a model plant. What is the mouse for the human studies is Arabidopsis for the plant studies. And it lives very happily in the Netherlands in a nature reserve. And when we do studies on the microbiome of plants, we use natural soil from a place where a lot of wild Arabidopsis is growing. And here you see students digging up the soil they bring it to the lab, and there we use the soil to study the microbiome and how it interacts with the plants. So how do we analyze microbiome? Maybe that's not known for everybody in the audience, so I'll explain it a little bit. So when we study the microbiome, so we first start with a sample, and we like to know what is the composition of the thousands of microbes that are in that sample. And then you can typically start with a sample such as from, let me see. Should be able to use pointer options. Then you see it? Sorry? Okay, well, anyway, then I will do it like this. You see the pointer? Yeah, the red dot, right? Okay. Yeah, so if you would like to analyze the microbiome, then you take a sample from, for instance, the soil, like here, this is the bulk soil, or you take a sample from the roots, all the microbes that live on the roots, or you can also take a sample from the gut, any sample where microbes are living, you take a sample, and after you take the sample, you amplify the 16S rDNA, which is typical for a bacterial species, and then you do next generation sequencing, and then when you do next generation sequencing, you get an impression about what is the composition of the microbial community in that sample. So for instance, if you do microbiome sequencing of the bulk soil, you get an impression about the microbial community in the bulk soil. And typically the bulk soil, here it is, the bulk soil often contains a lot of different microbes. There are like 10,000 of species are present in the bulk soil. And I show this in the type of post-stems. There are a huge collection of post-stems. There's a huge collection of microbes in the soil. But if you would look at the microbes in the rhizosphere around the roots, you will see that the roots selected certain microbes to the root system, and then there is much less diversity because the plants select certain microbes to the root system. Many of them are beneficial and help the plant to grow and defend itself better. So they only select the best, and in this case I show it by demonstrating that only the best post-stems are selected to the rhizosphere. I used every time the wrong button. Okay, so this is typically visualized like this. So on the left-hand side, you see the microbial composition looks like this. So there are many different types of microbes, but in the rhizosphere, you get a selection of certain microbes. There are much more of this gray ones and much more of these orange ones than in the bulk soil. So these are selected by the plants. So, and here you can also see that this really takes place. This is a typical microbiome analysis. On the left-hand side, you see many different spots, and all these spots represent the microbial community in a bulk soil. And they cluster together because they all look similar. Yeah. And on the right-hand side, you see a collection of microbial communities that are selected from the rhizosphere. They look very different than from the bulk soil, and that means that they are selected by the plant and that they have a different composition than the microbial community that is in the bulk soil. And how do plants select microbes to the root system? Well, they do that because they are able to secrete certain compounds into the rhizosphere. For instance, they secrete primary metabolites, such as organic acids, but they also secrete a lot of secondary metabolites. And the secondary metabolites have antimicrobial activity, and then they allow certain microbes to grow in the rhizosphere, and they do not allow other microbes to grow in the rhizosphere. And that's how they select their microbial community around the root system. And here we see an example of such a secondary metabolite that is produced by the roots. So here you see nicely a root of Arabidopsis in red, those are the roots. And inside you see a blue color, and the blue color are coumarins. And these are secondary metabolites, and they are produced in large quantities in response to certain beneficial microbes. And they are antimicrobial, so they inhibit growth of certain microbes but they stimulate growth of other microbes. And that is how the plant selects microbes around their root system that they would like to have and from which they can obtain benefits from. And while others are negative, they are inhibited. So if you zoom into the microbial community of the rhizosphere, you will see that there will be many, many Pseudomonas bacteria. That's a bacterial genus that really likes to grow in the rhizosphere. And here you see a circle, that those are genomes of 100 different Pseudomonas species. And if you look at the type of functions that they represent, you will see that they produce growth regulators, such as auxins. They produce defense elicitors that trigger the immune system of the plant. They produce nutrient mobilizers that allow more uptake of iron or phosphate from the soil. And they produce antimicrobials, which lead to inhibition of certain soil-borne pathogens. So they provide a lot of functions for the plants that are beneficial. Well, one of the strains that we use often in the lab is this Pseudomonas strain WCS417. And what you can see here is this is the strain growing on a plate, the WCS417. Here you see it growing in yellow on the root of a plant. And if it colonizes the root, you see a nice growth promoting effect. You see the root system becomes much more branched. This is with the bacteria. This is without the bacteria. And you also see that the root has become much longer. And you can imagine that this leads to a larger root system. It can take up more water. It can take up more nutrients. And this is how the plant stimulates growth of the plant, because well, the root system becomes bigger, and then also you can get up to 2-fold higher shoot fresh weight as well, and more yield because of the presence of the bacteria on the root system. It is an effect of the plant hormone auxin, as you can see here in yellow, because the auxin response is much stronger in roots that are colonized by the bacteria than roots that are not colonized by the bacteria. And most importantly, if you colonize the roots of the plant by these Pseudomonas bacteria, it also provides protection against certain pathogens, such as this Fusarium fungus. On the left-hand side, you see a plant infected by the Fusarium fungus, and on the right-hand side, it's also infected by the Fusarium fungus, but the roots are colonized by the Pseudomonas bacteria, so the immune system is much stronger and protects the plant against this fungal infection. Well, what we also see is that in response to these bacteria, the plants produce coumarins. As you can see here, they are fluorescent and they are abundantly secreted by the roots of the plant. And you can also nicely see that on this picture. I will skip this. So what we try to investigate is how can plants cry for help when they are attacked by a pathogen, and do they then start to recruit these beneficial microbes to the root system, so that these beneficial microbes can boost the immune system and provide protection against the pathogen infection. So we investigate cry for help from the root microbiome. And what we have shown is that if you have an infection with the downy mildew pathogen, which is a very important fungus on the leaves, that in response to that fungal infection, you get a change in the microbiome on the root system, and many microbes become recruited to the root system. And then in return, the microbes that become recruited induce the immune system of the plant, and then they provide protection against this downy mildew pathogen. So apparently the plant is able to recruit microbes to the root system that induce systemic resistance against the pathogen. Not only that, then the soil becomes conditioned with many microbes that are very beneficial. And this then also leads to the fact that when you grow a new generation of plants in that soil, that they are also better protected against the pathogen. So the microbes are recruited, they are more abundant in the soil, and then they provide protection also against the next generation of plants. For instance, the seeds that drop from this plant into this soil, they are better protected against the pathogen than when the bacteria would not be induced in the soil. How do we know this? Well, we know this because of this experiment. So what we do, we grow plants in pots, like we show here. We grow plants in the pots, and we infect half of the plant with the fungus. We call it HPA because that is the fungal pathogen. And if you have done that, you will see that the microbial community in that soil changes. And then if we remove those plants and we put the next generation of plants in those soils, then you will see that if you infect those plants with the pathogen again, that the plants that grow in the soil that was previously conditioned with diseased plants are less diseased. As you can see here, they are much less severely diseased than the plants that grow in this soil over here. So this soil contains soil from in which plants that were healthy were grown, and in this soil contains soil in which previously plants that were diseased were grown. And then the microbial community was changed, and that provides protection against the pathogen infection, as you can see here. Then, of course, the next question is, what are these microbes that are enhanced in that soil? And for that, we did a microbiome experiment, and again, compared the microbiome of unplanted soil on the left-hand side here, and we looked at the microbiome in the rhizosphere plants, and you can already see that the microbiome in the rhizosphere is very different from the microbiome in the unplanted soil, indicating that the plants select the microbes. But if you compare the microbiome of the healthy plants here in green with the microbiome of the diseased plants here in red, you will see that there's also a big change in the microbiome in the rhizosphere of the healthy plant in comparison to the microbiome of the diseased plant. And if you then zoom in, you will see that these are all the microbes on the healthy plants. You can do a network analysis of all the microbes on the healthy plants. So these are all different microbial species. And if you look at how they change in response to pathogen infection on the leaves, you will see that the green ones become more abundant, so they increase in numbers, and the pink ones, they decrease in numbers after infection of the leaves with the fungus. So indicating that the plant starts to recruit these microbes to the root system. And then you can zoom in. What are those microbes? Which ones become recruited to the root system? And then we were able to identify 3 microbes that really became abundant on the root system after infection of the leaves by the fungus. So that is a Stenotrophomonas, Xanthomonas, and a Microbacterium. You really see that they're highly abundant on the root system after infection of the leaves by the pathogen. And if you then select those microbes and grow them on a plate, combine them together, give them back to the plant, you will see that these 3 microbes, we call them XSM, if you apply them to the root system of the plants, then you will see that they promote growth. The plant's shoot fresh weight becomes bigger. You also can see that the plants become bigger, as you can see here. The control plants look fine, but the plants of which the roots were treated with the 3 microbes that were recruited, they grow much bigger, as you can see here on the right-hand side. But most importantly, if you provide the 3 microbes back to the root system, you get an enhanced resistance against the infection by the downy mildew pathogen, because you see much less severity. As you can see here, the plants are much less diseased when they are grown in the soil with 3 microbes in comparison to the controlled soil, indicating that the microbes that became recruited after pathogen infection, they provide protection against that pathogen that started the recruitment. So what I showed you up to now is that you have pathogen infection. This leads to a change in the microbiome. Certain microbes become selected to the root system. They become very abundant on the root system. And if you provide them back to the roots of plants, they stimulate the immune system, and then the plant becomes less diseased by the fungal infection. So the plant can basically recruit its own medicine in the form of microbes. Well, how does this work? I have not much time, so I will not talk too much about it. But one of the important components that is important for the recruitment of these 3 microbes are coumarins. And as you can see here you see plant roots of Arabidopsis. They produce a lot of coumarins in the fluorescent color. There's all coumarins. And we have demonstrated that the production of this secondary metabolite is very important for the attraction of the microbes to the root system. And then you would say, do the beneficial microbes, aren't they not harmed by the coumarins? Well, no. So the Pseudomonas that I talked about, Pseudomonas WCS417. It stimulates the immune system. And if you grow it in the presence of high levels of coumarins, in this case, scopoletin, high levels, 2 millimolar, then still the Pseudomonas grows happily. It is not affected by the coumarin. But if you grow a soil-borne pathogen, such as Fusarium, Oxysporum, or Verticillium daliae, these are very important pathogens in the soil. They are all very much inhibited in growth by the coumarins if they are produced. So on the one hand, the coumarins are important for the recruitment of the beneficials, and they are important for the inhibition of the pathogens in the soil. And well, with that, I come to the last slide. So I have shown that if you have a pathogen infection of the leaves, then this leads to the recruitment of certain microbes to the roots. They become very abundant on the roots. They stimulate the immune system of the plant and help to protect the plant against this pathogen infection. Not only that, the coumarins are very important for this selection. And well, when these soils are conditioned by these microbes that became more abundant in the soil, then also a next generation of plants that grow in that soil, they are better protected against the downy mildew infection. What can we do with this? Well, generating knowledge is extremely important, because if we know more about what these microbes can do, we may also make use of it in order to produce more foods in a more sustainable way. So 10 years ago, the American Academy of Microbiology investigated how microbes can help to feed the world. And they predicted that in 20 years, we would get a 20% increase in crop yield worldwide, by a 20% reduction of agrochemicals, by making use of beneficial microbes. And how can we do that? By selecting the right microbes, such as the ones that I talked about. We can make clever bioinoculants that we can, for instance, provide to the plants as a seed coat or as a spray, and that can help already to stimulate growth of the plant or provide protection against harmful pathogens. We can also breed our crops so that they are better able to select the microbes from the soil themselves. And we can also do better soil microbiome management, for instance, taking good care of our soil, because if you have a healthy soil, you have also a healthy crop, you have also a healthy product, and that's also healthy for the humans. And that is why I think research is very important, but not only research is important, also translation to application in the future will be very important. And with that, I would like to stop and thank you for your attention. And well, my whole group, they say hello to everybody here in Indonesia, and thank you very much.
Okay. Thank you very much. I am glad that we are started earlier, so we can warm up with the technical problems. And now we have 10 minutes earlier also for the Q&A session. So yeah, I would like to invite you to ask questions. You will stand over there, or you will sit beside me? Okay, sure. I know, because he's very healthy person. Okay, ladies and gentlemen, is there any of you want to ask question to Gordon? Okay, Ms. Rahmi, you can go to the front and oh yeah.
Thank you for the opportunity. Hello, Mr. Corney. It's such an interesting presentation, and I'm really interested to the topic. And actually, it's about the soil management. So, in your presentation, it's said that the soil that is being used for planting plant that is infected with some pathogen is better, provide better protection for the next generation of the crops. And what I'm asking is about what kind of soil management that is recommended, because I think in general in Indonesia, people still tilling the soil. So I think, and I heard it's not very beneficial for the root or bacterial in the rhizosphere. So what is your suggestion about it? Yeah.
Yes, well, that's a very good question because, yeah, around the world, of course, farmers do tilling of the soil after harvest. They plow the soil so that they get a good soil again for the next year. But what you actually then also do during the growth season, the plants that grow in the soil, they also select the right microbes to the root system. So after a growth season, this is basically in a very good condition, right? Like in nature, if you look in a natural situation, like in ecology or an ecological situation, plants grow always on the same place, and they nourish their microbiome, and they make sure that their microbial community is perfectly fitted to their root system. What we do in agriculture, every year we do something else. So we disrupt the microbial community, and the next year your crop needs to start all over again. Right. So for some crops that is important, because if you build up a lot of pathogens, like for instance in potato in the Netherlands, if you grow 2 years in a row potato, that's not good, because the pathogen is so important that builds up in the soil. No beneficial microbe can work around it. But for many plant species, it's natural that you have a good microbiome community, and then also for the next year that can provide benefits. So what, for instance, in wheat culture all over the world is now practiced, try not to till. So if you have no till, you can make use of the beneficial microbial community that was built up over the previous season in the soil, and then you would have better harvest in the next year, because the next year doesn't have to invest a lot of energy into recruiting all these microbes again, because they're already there. So no-till is, for many crops, better because you can make use of the natural microbial community that was built up in the season before. Yeah.
Okay. How about that? How about the answer?
Yes. Very much for the answer. Actually, I want to ask one more question.
Okay.
And
Go ahead.
so, what about the nutrition? I mean, how about the supply of nutrition? If we are not tiling, usually during with tiling, we enrich the soil with the nutrition. I mean, after a period of crop planting, the soil has deteriorated nutrition. So many farmer enrich their soil with fertilizer, whether it's chemical or natural fertilizer. And what about that for the microbial community?
Yeah. So the question is it on? Yeah. So the question is about. So what about the nutrition for the plants? So the fertilizer that are provided to the crops in agriculture. Well, I don't think that the microbes can completely replace the fertilizers that we produce, because if you harvest, you take so much nutrients from the soil that you need to refresh that also with fertilizers. But we use so much. If you look on the world scale, we use so much fertilizer, and much of the fertilizer are drained through the soil and never been used by the plants. So microbes can help to provide a better structure of the soil. They can capture better the nutrients that are in the soil and that are provided by the fertilizer. So if you reduce the fertilizer with a good microbiome, you might be able to get the same yield or even a better yield with a good microbiome with the reduction of the use of fertilizer. So I don't think it can replace all the fertilizers, but if you can reduce it already with 20–30%, I think that's already a good benefit also economically, because you need to use less fertilizers, you have a better microbiome. The microbes may help the plant to mobilize the nutrients and capture the nutrients in the soil and bring more to the plant than otherwise you just add fertilizer and they are drained, they flow away into the rivers and you never see them back again. So that's also not a good system. So I think they can help, but not to replace fully.
Okay. Also, I would like to invite one more question from the audience in online. Are there any question? You can ask directly and or using the or type your question on the chat box. Okay. It seems like there's no question in the online. I would like invite one more questions from the audience here at the venue. Oh, okay. Please introduce yourself first.
Okay, thank you for the lecture. There's very insightful lecture. I really appreciate that. Firstly, I would like to introduce myself. My name is Nur Alif Ahmad Mustafi, which you can call me Alif. I'm one of the bachelor student at biology education at Universitas Negeri Malang. And now I would like to ask you a question, simple question. If there are too many microbes present in the soil or the plants, is there any possible negative effects for the plants, or it just strengthen the plants? Thank you. I really appreciate that.
Thank you. So your question is, if there are many microbes in the soil, if it's better for the plant than less microbes, all right? Yeah, well, more is not always good, but a plant can only select from the microbiome that is present in the soil what is there. If it's not there, it's not selected, of course. So good microbial diversity in the soil is always good, because then the plant has more to choose from, right? So because let's say the plant roots, they select 10, 15% of all the microbes that are present in the soil. And so it inhibits some others so they don't grow, and it stimulates, let's say, 15% of all the microbes that are there. So if there are many beneficial microbes in the soil already present, then there's much more to select from. So a good microbial biodiversity is very good. If you reduce the microbial diversity because of tilling or use of agrochemicals, because if you have a lot of nutrients in the soil, also not good for the biodiversity, because then only the microbes that like all these pampering with nutrients, they like it, and they grow very abundant. And microbes that are also but they don't like too much nutrients, they don't grow. So the microbial biodiversity is very important, because it gives a good starting point for the plant. But not all microbes are good. There can also be bad ones in there. But in general, a good biodiversity is better for the plant because it has more to choose from.
Okay. I think because of the time, the limitation of the time, we need to finish the Q&A session here for the first session ever of the fifth iFollis 2022. And before we start, we continue to the next speaker. I would like to invite Cornel Peters and also the Dean of the Faculty of Mathematics and Natural Sciences to give the merchandise. Like a stand of merchandise.
Nag-feedback pa
Okay, without further ado, I would like to introduce you, our second keynote speakers for today. Okay. Our second keynote speakers for today is coming from the Philippines. But before that, I would like to mention that I will never forget the first time Malang was locked down because of the spread of COVID-19 in early 2020. I didn't forget about that because in the early of 2020, my son was born. So my son was born basically 2 weeks after lockdown. So we only have a limitation to go to the hospital, something like that. And yeah, all aspects of our lives becomes difficult. At that time, many rumors about COVID-19 were initially found in bat in Wuhan City in China. So the possibility of disease from animals to human is quite high, and we call it the zoonotic diseases. So today we will hear the remarkable presentation from our keynote speaker, Professor Ma Asuncion Guiyang Beltran, PhD. She is an animal husbandry and veterinary medicine expert from the College of Veterinary Medicine, Tarlac Agricultural University, the Philippines. She graduated from a master program in tropical veterinary medicine at James Cook University, Australia, and in animal husbandry at Gregorio Araneta University Foundation in Manila. And she took her PhD in animal science at Central Luzon State University. Currently, she is the Vice President of Research, Extension, and Training at Tarlac Agricultural University. She has also become an associate member of the National Research Council of the Philippines in 2017 until now. She is an active speaker in many seminars, conferences, and training internationally. Since 2020, she has been actively doing a project transforming backyard chicken production into a viable enterprise until 2023. So, ladies and gentlemen, please welcome and give big applause to our keynote speaker, Professor Ma Asuncion Guiyang Beltran, PhD. How are you, Professor Beltran? Can you hear me? We still cannot hear your voice.
Oh, okay. Did you hear me now?
Yes. You have 20 minutes to deliver your presentation and then followed by 10 minutes for the Q&A session, Professor Ma.
Okay. For a while, because okay, so my topic today is about One Health approach for zoonotic disease, and I am Dr. Maria Asuncion Beltran of the Tarlac Agricultural University in Camiling, Tarlac in the Philippines. So we all know that One Health is recognizing that the health of people is connected to the health of animals and their environment as well. So One Health approach is by working out with physicians, veterinarians, and ecologists to monitor and control public health threats, especially zoonotic diseases. And of course, to encourage the collaboration efforts of a multiple discipline, okay, we have to be working nationally and globally to achieve the best health for people, animals, and our environment. So this is good slide that, I'm sorry. Okay. Is my slide clear?
Yes, it's clear.
Okay. So this is an umbrella of One Health, which is there is a collaboration between animal health, human health, and of course the environment as a whole. Of course, there's a lot of things here, especially if we talk on zoonotic infection. Of course, with human medicine, the comparative medicine, and translational medicine. Of course, we do a lot of topics here on environmental health, on ecology, on veterinary medicine, public health, human medicine, molecular and microbiology, health economics, with individual health, population health, and the ecosystem health as a whole. So, in order to have a very good, healthy One Health, this is a triology system. We must have healthy people, healthy environment, and healthy animal in order to have a very good One Health. So this is it. This is the illustration. And of course, we know that Earth is not getting any bigger. And because of overpopulation, yeah, we have pandemics and there are a lot of things that happen for the past 3 years. But because of this one, Earth is not getting any bigger. So the difficulty of maintaining One Health is really difficult when a lot of things could be intermingling with each other. Okay, so why One Health is important? It is an approach. It is very important because 6 out of 10 infectious diseases in human are spread from animals. This is according to CDC in 2016. For instance, some diseases can be shared between animals and people, and these diseases are called zoonotic disease. Okay, so the cooperation achieved will progress healthcare during our time and beyond by advancing biomedical research, enhancing public health safety, and improving medical, veterinary education, and clinical care. And of course, not to mention very good environmental care. Okay, so successful public health intervention require the cooperation of the three, the human, animal, and environmental health communities. And by promoting this collaboration, we'll achieve optimal health outcomes for both people and animals. Okay, so we all know that this is the epidemiological triad. Okay, the agent could be, of course, everything: bacteria, virus, parasite, fungus, mold, even chemicals, radiation, heat, natural toxins. The host, of course, it could be human or animal that harbors the disease. And of course, the pathogen. These are the disease-causing microorganism, or could be the agent also, the related substance that could offer subsistence and lodging for the pathogen. And of course, we also look on to the level of immunity, the genetic makeup, the state of health, and the overall fitness within the host, which could determine the effect of the disease organism that could have upon it. So we all know that there must be, in order to have that epidemiology, we have this called triad. Okay, this is it. Of course, when we say on the environment side, we have to take good care on the temperature. We all know that temperature is going high now because of the climate change, humidity, altitude, okay, and of course, crowding, especially on animals, housing, also neighborhood, water, milk, food, radiation, pollution, and noise, and of course, the biological agents. Okay, so what are some factors that increase disease outbreak? Okay, human population, number one, are growing. Of course, especially in Asia. The number one continent that increases populations is in Asia and in Africa. And the Earth has experienced changes in climate and land use, such as deforestation and intensive farming practices. International travel also. Sometimes the mosquito that causes disease in Africa could be traveled through plane because of international travel and trade have increased. These are some factors that increases outbreaks of diseases. Of course, the fast increase in human population, urbanization, and travel enables sustained transmission of human diseases. Okay, also the Asian lineage of chikungunya virus originally circulated between monkeys and mosquitoes with spillovers into human, but recently evolved in human mosquito-human transmission cycles, which results in epidemics. Good that we don't have chikungunya yet in the Philippines. So there are reported drivers, including human migration, settlement of mosquito vectors and urban ecosystem, and increased farming activities near the forest. Okay, so also the population of rodents that could carry a range of viruses and are abundant throughout the world. It also accounts about 40% of mammal species. They occupy also a wide range of habitats and reproduce in high rates and thrive on contaminated food and water. Okay, sometimes we consume even rodents. At least fifth of the world's grain output sometimes are eaten by rodents. And also another type of disease are the Hanta and Lassa viruses that have emerged as a major causes of zoonotic disease. And it could survive rodent excrement, and the aerosolized infectious particles and dust also could infect humans. Okay, deforestation is also one. It's because of these, the emergence of hinder viruses and the destruction of bat habitats. You know, bats are number one vector sometimes of diseases coming from those animals in the forest, and the increased contact between some flying foxes and humans, decreased migration also, which in turn lead to lower immunity in bat population, and of course, increased virus circulation. So the Hendra virus infects also horses and human, causing respiratory diseases in both encephalitis in humans. So the first outbreak of Hendra virus in 1994 in Northern Australia, and it has since had recurrent small outbreak and is identified in southern coastal of Australia. So the international trade also of pigs, but because of the African swine fever infection, it lessens already the causes of some disease when it with the spread of the ASF virus. But good enough that we don't have already some of the pigs, especially here in the Philippines, because of the increased infection of the African swine fever. Of course, the global trade also of live animals, which includes our small and large ruminants, particularly in the Central Asia and Africa, that sometimes because of our illegal imports, the foot and mouth disease contaminated food items makes FMD epizootic, and there have been great occurrence of the disease in different countries. So more people live in close contact with wild and domestic animals. So that is one very good risk factors in having a zoonotic disease. So a disruption of environmental condition and habitats provide new opportunities and disease to pass to animals. So with the more contacts with animals, because bats sometimes are the most popular reservoir host of several viruses and could pose some health risks to human, including SARS-like coronaviruses, Nipah and Hendra viruses, Ebola viruses, rabies viruses, and related Lysa viruses. And these are the factors that contribute to bats being a reservoir. It include high species diversities, long lifespan, ability to engage in long distance movement and dispersals because they fly really long, and the use of the torpor and hibernation, and factors related to host cell biology could contribute also to the risk factors of zoonotic diseases. Because of the disruption of the animal habitats, they will invade the farming landscape of human and living environments. Bats are almost seen everywhere in the world. And of course, we know the COVID viruses have been spread first by bats in China. That's why we have the COVID infection that started in China and spread all over the world that becomes a global pandemic. So the encroachment of human into the natural habitats of monkeys may result also in increased pathogen spillover to human. So we give also a progressive exposure of human to the new pathogen. So it generates also species jump with sustained human to human transmission. So we all know that 60% of existing human infection diseases are zoonotic. So 60% are zoonotic. That means diseases of human coming from animals. So 75% of emerging infections are diseases of humans, okay, which have been from animal origin. And 80% of agents of potential bioterrorist use are zoonotic pathogens. You all know that we got a lot of some potential bioterrorism attacks coming from agents, and there are 80%. And 5 new human diseases appear every year, and 3 are of animal origin. This is according to CDC. So I will just give you a rundown of the most popular zoonotic diseases that we have, not only in Indonesia, not only in the Philippines, not only in Asia, but the whole world. Okay, example are the Nipah virus infection, rabies, bird flu, Ebola, salmonella viruses, SARS, of course. We have the COVID-19, okay? The most recent that created a lot of scare and a lot of death among those affected one. And of course, we are so very nervous about the different strains that coming out almost, twice a year. I have been affected with COVID-19, I myself, and, I was just recovered from it. And this example of Nipah virus, it's the same family out of the COVID virus. And it looks like this, And of course, Nipah infection is a newly emerging zoonotic disease, and it is a severe disease from both animal and human. And of course, the natural host is the fruit bat, and they are triggered by deforestation, directing fruit bats, and the rest. So, of course, I'll just give you a rundown of this one. This is how it goes, and this is how it spread. And of course, rabies is the next one, and it infects the central nervous system, and it causes the untimely disease in the brain and death. And of course, the most commonly affected are the man's best friend, which is the dog and the cat. Of course, it will give you a symptoms of just like, rabies, and human is like rabies, and people also that includes fever, headache, general weakness, and discomfort. And this one, death usually occurs within days from the onset of symptoms. Of course, there are a lot of nervous symptoms that will be appearing if we have rabies, like that one in red. It will give us hallucination, hypersalivation, hydrophobia, etc., because it usually affects the brain. So it started first, the virus entered into the body through animal bites, and then the virus replicate into the muscle, and then it will go to the peripheral nervous system, and it replicates into the root ganglion, it travels into the spinal cord, and then until the brain is already infected, and the virus travels from the brain into the different parts of the body, like the eyes, the kidneys, and the salivary glands. So this is how we control it. We usually do vaccinating our dogs, and we all know that 40% of the victims are children younger than 15. That's why we have to prevent the transmission of rabies, particularly in dogs. And of course, we have to learn about this because this is, okay, another popular zoonotic disease is the bird's flu. And bird's flu or avian influenza is a viral infection also that spreads bird to bird. But of course, there is a deadly strain, the H5N1, which continues to spread among poultry in Asia, and not only one, it's all over the world. It's a highly pathogenic avian influenza, or the H5N1 or the HPAI, deadly to most bird, but it could affect also human being. So it could be coming from the migrating waterfowl, and because of the big spread to birds, pigs, and even donkeys, it spread firstly in 6 nations: Bangladesh, China, Egypt, Indonesia, India, Vietnam, and it keeps on recurring again. So this is, okay, if I could go back. Okay, and these are the infection. It's a rare event in human, but it appears, okay? It appears also that we could have to start influenza in human through the avian flu. Another one is the Ebola virus. Of course, Ebola disease or the Ebola hemorrhagic fever first appeared in 1976, but it is an outbreak of acute viral hemorrhagic fever in Africa. And then it already have an outbreak in 2013. And then the Ebola virus, it goes along with the genus Marburg's virus. And then it includes also a lot of species already. And it has been responsible for the outbreak in Africa. That's why it is observed also in Asia, a human disease caused by the Reston Ebola virus. Of course, fruit bat is the one responsible also in spreading the disease. Okay, and of course, it causes also a decline of the African chimpanzee and gorilla population in the recent decades. So Ebola virus is transmitted to human through close contact with blood and bodily fluids with another infected human. So it could either be direct contact or indirect contact from the contaminated environment. So of course, we have to do a very good healthcare. This is showing the Ebola disease virus in West Africa, and the lower part of the slide is the appearance of the Ebola virus taken under the microscope. So this is how the Ebola virus goes into the human infection. Ebola hemorrhagic fever, of course, and then you got the signs there, a lot, rash, red eyes, hiccups, until bleeding inside and outside the body, difficulty of breathing, vomiting, stomach pain, and of course, the transmission could be direct or indirect. And the prevention, of course, we don't have, I don't know if we have virus already, but of course, if you are there on the area, we've got a lot of infection, we have to do a lot of prevention. Okay, and of course, salmonellosis is one very popular zoonotic diseases. This is one scary disease in the US coming from infected poultry products. So it's a foodborne disease caused by Salmonella bacteria. And because of the consumption of contaminated food from usually meat to poultry, poultry eggs, and milk. It affects the gut or the digestive system. And of course, we need an immediate medical treatment for these. So it comes from water, but sometimes reptiles and amphibian carriers are recognized also as risk factors, and transmission could also be transplacentally. And 20 bacteria may produce 9 typhoid gastroenteritis also. Of course, these are some pictures on having salmonellosis infection. It progresses within about 12 to 72 hours and until 1 week. It ranges from a mild to severe infection, and treatment could be antibiotics orally and injected for 2 weeks. Okay, so this is a very common infection, not only in Asia, but all over the world. So, of course, we are veterinarians and most of you are biology. So, of course, when animals are healthy, we have to give them vaccination. And of course, animals usually have this one, Salmonella dublin, Salmonella typhimurium. Okay, other Salmonella causes also abortion and causes dead cows and calves, and there is a scouring effect on small animals. Of course, the next one, next zoonotic disease are, of course, the SARS virus earlier than our COVID infection. So it looks the same because it comes from one viral family. So it started in 2002 in China, and they got a lot of deaths with 8,000 almost affected one, but it results in 774 deaths. And of course, the natural bat virus is also part of it, and also camels. And because of this, it reaches human being and they are the amplifier host. Almost the same with the COVID-19 infection. Of course, the whole world stops 2 to 3 years ago. These are the first year that we meet our classes face to face. This is already on the last second quarter of 2002, but still there are some more infection very active in some Asian countries and all over the world. And of course, it is caused by novel coronaviruses, and it circulates among animals, and of course, it affects also human. These are the symptoms that we have. We are all familiar with this one. It started with cough or fever, and then we will have a difficulty in breathing, muscle pain, tiredness, and of course, we got a lot of prevention when all of us have already masked, and the contact with each, we need to have 1 or 2 meters apart. Okay, and the whole world developed these droplets that causes infection to other people. Okay, so if you could see the different viruses in here, this is the novel coronavirus, a very small one. If you could see on the lower part, that's why it's very difficult. If you could see the slide, and this is the history of the pandemics in the world. Okay, so we got the Spanish flu, 40 to 50 million died, Russian flu, the corona outbreak, and then the HIV/AIDS, and it's still ongoing. 25 to 35 million is affected, the Asian flu, the Hong Kong flu, and these are also the date. You see, 1981 to present, 2012 to present, the MERS-CoV, the Ebola until now, of course, swine flu, and now the ongoing COVID-19, which started in 2019 until the present. Okay, so these are the viruses that causes zoonotic disease, and because of the size of the viruses, they could be easily transmitted to different human and different animal. So, yeah, who are not scared of coronavirus. And of course, we got a very good control thing that starts from the community, and then that start from each one of us, and then the productive equipment, we use gloves, and all of us are now in mask, and the administrative control of our government, the engineering control also, substitution. And of course, I hope and pray that we'll be eliminating the virus as soon as possible, because we need to contact with each other. Nowadays, we need to partner with each other. And of course, be ready with another surge if things will be uncontrolled. WHO is giving us advice. The World Health Organization is giving us advice on how to protect ourselves and others. So we have to be safe. We have to be smart to inform ourselves and be kind to support each other with the COVID-19. So to ensure One Health, to ensure our security and stability with the COVID-19 infection, of course, we have the government's governance and leadership with each of the country. Of course, we got 5 areas in order to control the COVID-19. We need to have a surveillance and monitoring, and we need to contain and control the disease in each of the respective countries. Of course, we have to save lives. We have community resilience, and we have to do security and stability. And this includes all parts of the society. We do research, we do recover, and we do reflect in order to have and maintain One Health, not only in our respective countries and the whole world as a whole. So as a veterinarian, if I may say, because I am a veterinarian, we need to have a day one competency. I will not be discussing a lot of this one, but when we say day one competency, we are responsible in promoting animal health, animal welfare, veterinary public health, and food safety at day one after graduation, because we are part on doing animal health. With these, I may get all your time already. I'll just be around none of these. We need these competencies, the knowledge, skills, attitude, and aptitude in all professionals, not only veterinarians. In order to have a day one competency, we must be ready in our respective profession to have our competencies in our different knowledge, skills, attitude, and aptitude. So with these, we need to have a basic, clinical, and advanced competencies, specific competencies, advanced competencies. And with these, I would like to thank all of you and listening to my short lecture. Thank you very much.
Yeah, thank you very much, Professor Maria Asuncion Beltran. So there's already one question from the chat box. I would like to read for
Okay.
you.
Yes.
The question is from Agik from Airlangga University, Surabaya, Indonesia.
Yes.
So some animals are used to help research, for example, rats and rabbits. Do you think these animals can also pose a threat to the spread of disease to humans, especially researchers? Please explain.
Okay. We have lab animals. Okay. Like, for example, your rodents, your white mice, your rabbits. Okay. And they have to be maintained in a very good laboratory. Of course, if you maintain that in a very good laboratory, you have to, you can control also the spread of the disease. So these lab must be contained and must be sustained and must be maintained. in order not to spread the diseases that you are researching in that particular laboratory.
Okay, I think that's quite clear for the answer. And I would like to invite one more question. Okay, please. You can please give the microphone.
Yes. Yeah. Thank you. Magandang umaga.
Wow.
Already. Ang pangalan ko ay Anita.
Yes, Anita.
Yeah. That's mean my name is Anita. And I was also taking my master in James Cook University. So I feel like I have you as my friend. And my question is how to prevent the zoonotic diseases like what you said, and who is the most risk for getting zoonotic disease? Yeah, maraming salamat.
Okay. Of course, the number one risk factor are human beings. Like what I have lectured you, of course, we are starting invading our forest, where animals, particularly more on bats, which is the number one reservoir of some of our diseases. That is number one risk factors. And of course, we have to take good care also of the environment. We have to maintain also of our environmental health. So that includes on how we, by the way, I am with my students. All of my students in my environmental science are attending your international conference. So hello, everyone.
Hello.
Maybe my student
Hello,
could raise
everyone.
their hands and say hello. Yeah, hello, everyone. Yes, yes. Of course, the number one risk factor are human being. Of course, with our production system and with our on how we control our animals if we are invading the forest where they usually, where you usually live. And of course, with our international travels also, and sometimes people doesn't have discipline enough. If they got already these diseases, they keep on roaming around without observing the right thing to do. Of course, we have to be disciplined enough just to control the diseases that are, especially COVID-19 nowadays. If they said we must have proper distancing, then we have to observe that. And if there is a regulated face-to-face, then we have to be regulating face-to-face. That's why I really wanted to come and attend the conference face-to-face, but so we got some regulations yet. So of course, the number one risk factor are the human being, because we have the brain to control it, but sometimes we are not doing it. So that's it. It is the environmental health, the animal health, and at the same time, human health has to be in congruence with each other in order to maintain one health.
Okay, thank you very much, Professor Beltran. So, unfortunately, due to our limitation of time, we need to continue. And I would like to say hi to all of you in the Philippines. Hello.
Okay,
Welcome to ICOLIS. Welcome to ICOLIS.
Yes.
Thank you for watching our international conference. And we very honored to have you as our speaker in this year. And we wish you here actually, to know you more in person, but it's okay. Since we have an online platform, we can do the hybrid conference. Thank you very
Okay.
much.
Thank you very much.
Yeah, on behalf of the
Last
committee.
way.
Goodbye. Thank you very much.
Goodbye. Thank you very much.
Okay, ladies and gentlemen, that is our first plenary session for today. We will have 10 minutes for break. So after that, you will need to come here again at 10:15. So my name is Fauzi. I'm from Biology Department. I do apologize if I have mistake to mention names to tell you something that not acceptable. I'm so sorry. So without further ado, I would like our session. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
All right. Thank you, Mr. Fauzi Akbar. Well, that was our first plenary session from Dr. CMG Corner Peters from Utrecht University and Dr. Ma Assumption Kubiltern from Talak Agricultural University. Thank you for the knowledge you have shared. And for the next agenda, here we have the first break. So for all the participants, I would like to remind you that the break lasts for 15 minutes, and at 10:15 a.m. is the time for the second plenary session. For participants in the Zoom, you may not leave this room, and you can turn off your camera. Thank you. Baik teman-teman, saat ini kita memasuki sesi coffee break. Teman-teman dipersilahkan untuk beristirahat selama 15 menit dan kembali ke tempat ini pada pukul 10.15. Terima kasih.
Welcome to Universitas Negeri Malang, UM. Located in 3 different places, UM provides a comfortable environment for teaching and learning. The main and second campus are located in Malang, surrounded by educational facilities, places for recreation, sports, and business. The third campus is located in the same area as the Shrine of Sukarno, Indonesia's first president in Blitar, East Java. Established on October 18, 1954, UM was born with the name of University of Teacher Education, or PTPG. In 1957, it became the Faculty of Teacher Training and Education, or FKIP. Then, in 1963, it became the Teacher Training and Education Institute, or IKIP. Finally, in 1999, it transformed into Universitas Negeri Malang, which is known to this day. Since its establishment until now, UM has established itself as one of the 12 educational higher education institutions and is included in the 15 best campuses in Indonesia. One of the visions and missions, UM, is to become a leading university and an authoritative center of excellence in the fields of science, technology, and education. develops a flexible, adaptable curriculum taught by teachers who guide students and provide skills for the future workforce to become active lifelong learners, independent problem solvers, creators of better communities, and bring education to life. educates students across 8 faculties. Also conducts special programs such as postgraduate program and teacher professional program. Has been granted an unggulan or excellent by the National Accreditation Agency of Universities or BNPT. Furthermore, there are 4 study programs that have been assessed internationally by AUNQA, and more study programs are accredited by ASIIN and ACUS International Accreditation. As one of the reference universities and a center of learning innovation, continues to develop knowledge and scientific research by providing quality education and training. has also produced many research products that have the right patent, published internationally, and ready to be commercialized. In line with our new tagline, Excellence in Learning Innovation, is the house for top science and technology center in higher education in DLI, or Disruptive Learning Innovation. Disruptive Learning Innovation Center develops innovation in smart learning with research focus on digital transformation on education ecosystem, either in higher education, school, or community, to improve the quality of education service in Indonesia. Many invention and implementation of products resulted from DLI research are expected to support continuous, innovative learning activities. To support the activities of residents in carrying out their daily duties on campus, provides unique and interesting learning environment through great and comfortable facilities to facilitate both academic and non-academic activities. has also made collaborations with many partners in the form of work training in education, research, and community service. Growing rapidly in this millennial era, has the vision to be a higher level institution and a reference university in the field of learning and education. has always been ready to dedicate itself to be the best university by producing the best human resource. The only one. Excellence in Learning Innovation.
It's here with us again.
Yeah, come on.
What? It's a CAKRAWALA.
Mindset University. People say that the world won't orbit around you, 'cause it's so hard to look up when you're feeling down.
Ain't nothing in this world, paper, just how you like it. So don't stop, won't stop till you make yourself proud. And just keep on moving. Nothing's gonna stop us now. Let's prepare our future. Nothing's gonna waste. Let's make our way brighter. Gotta let it shine. We'll be up in the sky. Come on, baby, step inside my world. See how now we're turning and won't stop. 'Cause we ain't dreaming, we make it real. It's time to make a change. See, you're trying to get the #1, but don't
don't forget what you want, keep your hope high. Make every time count. Get that confidence, build up and know what you are for. There'll be bumps on your way, cause perfection is so quick to pour. Nothing's gonna do it if we ain't. The art of achieving is when we are learning all day, learning all night. We'll shine, and you'll see me rising. Just keep on moving. Nothing's gonna stop us now. Let's prepare our future. Nothing's gonna wait. Let's look our way brighter. Gotta let it shine. We'll be up in the sky.
As the world keep moving, people will always keep changing. So stop daydreaming about your broken heart. Set your goal up high, then you'll see the light shining brighter than the sun.
Let's clap together and sing along.
Come on, baby, step inside my world. See how now we're turning and won't stop. 'Cause we ain't dreaming, we make it real. It's time to make a change.
Come on, baby.
Come on, baby, step inside my world. See how now we're turning and won't stop. 'Cause we ain't dreaming, we make it real. It's time to make a change. Come on, baby, step inside my world. See how now we're turning and won't stop. 'Cause we ain't dreaming, we make it real. It's time to make a change.
It's time to make a change. Come on, baby.
Seiring berjalannya waktu, semua bergerak cepat, semakin tak menentu. Seiring berjalannya waktu, semua berubah cepat, penuh tantangan baru. Kami pun terus berpacu, jaga dan meningkatkan mutu, tiada henti berinovasi, raih prestasi, banggakan negeri. UN berkarya dan akan terus berkarya, buktikan ilmu membangun nusa dan bangsa, menjadi unggul rujukan untuk semua. Excellent in learning innovation, excellent in learning innovation.
Seiring berjalannya waktu, semua berubah, semakin tak menentu.
Kami pun terus berpacu, jaga dan meningkatkan mutu, tiada henti berinovasi, raih prestasi, banggakan
the only one. Satu tekad untuk mencetak generasi yang cerdas, unggul, dan berdaya saing.
Berkarya yang akan terus berkarya. Hati yakinmu membangun nusa dan bangsa. Menjadi rujukan untuk semua. Ayo semuanya ikut berkami. Let's sing together. One, two. Berkarya dan akan terus berkarya. Hati yakinmu membangun nusa dan bangsa. Menjadi rujukan untuk semua. Excellence in learning innovation. Excellence in learning innovation. Excellence in learning innovation. Excellence in learning innovation.
Dari Iconic 2022 untuk segera kembali ke aula sekarang juga. Saya ulangi, dimohon kepada seluruh tamu undangan dan juga peserta Icolis 2022 untuk segera kembali ke tempat karena acara akan segera dimulai kembali. Terima kasih. Kami ingatkan sekali lagi kepada seluruh tamu undangan dan juga peserta dari Icolis 2022 yang masih berada di luar, dimohon untuk segera masuk kembali ke dalam karena acara akan segera dimulai.
Sekali lagi diingatkan kepada para peserta dan tamu undangan diharapkan untuk masuk ke dalam ruangan, dan untuk para peserta dan tamu undangan yang berada di ruang Zoom segera kembali dan mengaktifkan kamera. Ladies and gentlemen, the next agenda will be started. Please come back and turn on your camera. Ladies and gentlemen, the next agenda will be start in a minute. Please come back and turn on your camera.
All right, ladies and gentlemen, now we step to the next agenda, which is the second plenary session. Here we have Dr. Reti Retnawati, an Indonesia Psychological Society and National Commission on Violence Against Women, and Dr. Deka Sermwitawayong from Prince of Songkla University as the keynote speakers, and also Mrs. Nani Farida, SI, MSI, PhD, as the moderator in the session. For Mrs. Nani Farida, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay. Good morning, ladies and gentlemen. Welcome back again to the International Conference of, I call it 2022 in this second session. Okay. In this session, my name is Nani Farida, and I will be serving as your moderator. And in this session, you will be hearing two presentations from, the first one is from Dr. Reti Retnawati from Universitas Brawijaya, and the second one is from Dr. Dek Sermwitayawong. I hope I pronounce your name correctly. From Prince of Songkla University, Thailand. Good morning, Dr. Reti and Dr. Deka. Good morning, Dr. Reti and Dr. Deka. Are you
Morning.
in? Okay, good morning, Dr. Deka.
Good morning, I'm in.
Okay, great. Nice to see you all. Okay, before these presentations begin, let me kindly remind you again of the regulation in this session. First, I would like to give the time to Dr. Reti Retnawati to deliver her presentation for 20 minutes, followed by a presentation from Dr. Deka Sermwitayawong for 20 minutes as well. And after that, we will have a discussion with both speakers for about 20 minutes. Starting from now on, all participants online, you can start typing your questions for discussion on the chat room, mentioning to whom the question is addressed to. But if you like to ask directly to the speakers later on, you are welcome as well. Okay, before we start the first presentation, I want to read the summary of our first speaker's biography today. It's Dr. Reti Retnawati, MSc, IF. Is a lecturer in the Department of Physiology, Faculty of Medicine, Universitas Brawijaya, Indonesia. She is also a commissioner in National Commission on Violence Against Women, and also she's very active in Indonesian Physiological Society. She has received a bachelor degree in medicine from Universitas Erlangen.
I cannot hear you, Ibu Nani. Must be something wrong with my connection.
I can't hear the speaker either. I think she was the person.
Halo. Halo, Ibu Nani. Tidak bisa mendengar ya?
Hello, check Bu Reti, can you hear me?
Sudah bisa.
Halo, Bu Reti?
Ya, sudah bisa didengar suara saya?
Sudah, Ibu.
Oh, okay, sorry. Thank
Okay,
you.
Bu Reti.
Yeah.
Okay, Bu Reti, you will have 20 minutes for to deliver your presentation.
Okay. Thank you, Bu Nani, for the to be the moderator and mention some of my CV. This is my topic: gender-based violence from cell to the community. Next slide, please. As mentioned that I actually in sorry. First of all, I would like to present that the what is Komnas Perempuan. Actually, Komnas Perempuan is the National Commission on Violence Against Women, and this is one of the national human rights in Indonesia. So we have the special accountability that the annual and anthem of the response are submitted to the president, and we are actually an independent body and one of the national human rights institution. So the other two is KAHAM and also KPAI for children. I'm the commissioner in the priority issues in 2020 to 2024. Next slide, please. And so we have some tasks here to promoting the public awareness and to conducting research and review, monitoring the fact-finding and reporting the violence against women and women's human rights, providing input and recommendation to the executive, legislative, judicial branches of the government, and also developing regional and international cooperation. So we have 15 commissioners, and we have 12 women commissioners, 3 men, and 2 are the person with the disability. Next slide, please. So we come from different professional and academic to fulfill the Paris Convention as national human rights institution. Slide, please. So just to give you the idea, this is one of the data collecting by the Komnas Perempuan and also the Mitra and also the Badilak. If we look at the 20 years, actually when the gender-based violence has increased from 3,000 to 400 something thousand. So this is the increment is very high, and could be because of the people who'd like, I mean, exactly the victims that like to report, but this is the thing that been reported. We believe that is underreported is higher than that. So this is as the iceberg phenomenon. Slide, please. So in 10 years, actually, the gender-based violence, especially to women, it that we're supposed to be 2,240,070 something. And so if we divide it in a year, we will have the 224,7959. In a day, we have 624. In an hour, we have 26 people as victims of the gender-based violence in Indonesia. Next slide, please. We have 10 provinces with the highest GBV here, from Jawa Barat, Jawa Timur, Jawa Tengah, Sumatera Utara, DKI Jakarta, Sulawesi Selatan, Lampung, Riau, Sumatera Selatan, and Sumatera Barat. Next, please. And this is also, we have a higher, the type of the gender-based violence here is in the left panel, we have the physical, and then psychological, economic, and the sexual. So this is the number that's in 1 year last year, the data collected last year. Next, please. And if we look up to the relation between the victims and the perpetrator in the personal relationship, there are, most of them are known, the close person in the house or in the relation between the victims and the perpetrator. So this is, the myth is that the perpetrator is outside your house or outside your relationship, but that is not true. Slide, please. In a cyber-gender-based violence that we recognize that is increased from 2017 to 2021, from 16 to 1721. So this is the increment also, actually also influenced by the pandemic for the 2.5 years. So this is the increment. So people cannot go anywhere, and then they stay home using the handphone, and also the perpetrator stay beside them. Next, please. With the disability, especially the women with disability, also increase, especially in the intellectual disability. So this is the number is quite higher, but again, this is the data can be collected, but if we do not have any report, then we don't have the number on it. Next slide, please. And what we call, if an education environment from university or the vocation, we also have this sexual harassment. So this is, if we compare to the from 2015 to 2021, there is the increase of the number. But if you look up in the types of the institution, the highest is from university, and then the pesantren, this is the religious school, informal, and then high school, and then followed by the junior high school, and then the vocation in the others until the okay, next slide, please. And unluckily, actually 174 cases that been found by Tirto in 2020. They only 29 victims can be briefed to report to the campus, and this is very small number. What is the impact for the next slide, please? The negative impact of this violence, gender-based violence, actually physical and psychological health problem, changing in social behavior, economical problems, human rights problems, and education problem, or also attempted suicide. And temptation that is usually caused by the psychological problem here. Next slide, please. So what is can be seen as a decrease in tuition and learning motivation, low self-esteem, high tendency to skip school, low achievement academic, mental health problem, anxiety and depression, post-traumatic stress disorder, social relation problems, self-injury, pregnancy, suicide, and could be psychosomatic health problems, low psychological well-being, and also low self-confidence. Next slide, please. This is the positron emission tomography that is saying PET, we call it, that can detect the cell that in the brain that have the metabolic activity 100%, and show by the red color in here compared to the others that is from abused children, that has the dark, and here is a black. So the activity of the metabolic cell in this area that known to have the function as the intellectual function and also the emotional function. So this is very old, actually old photo from the PET taken in Romania in 2010 or something. Next, please. So there is some studies that is trying to look at what is actually the main risk, the main negative impact is depression actually. In Kenya to 2018, that the if the victims less than 16 years, then the depression will, the light to mild depression will occur in 14.6, and 85.4 is mild to severe depression. But if the victims age more than 16 years, the depression light to mild is about 6.4, and the other 93.8 is in mild severe depression. In South Ethiopia, for example, in 2020, they said about 59.6% is in depression. And then in Cambodia, studies in a female entertainment worker, they have about 65.9%. So as we can see here, that depression is the most common to the victims. The next slide, please. And then if we look up in the function biologically, actually the depression is in the brain. Then the brain will produce several hormones and some molecular level things. And second, we have to realize that there's all the stimuli come out from the body, goes to the brain, that will influence all the activity of the brain and the other nerves around that. Next, please. So we realize that there is the brain and the nerves from sympathetic and parasympathetic here. They will give the nerve in the eyes, for example, salivary glands, lung, heart, the stomach, pancreas, liver, and also the bladder and genitalia. So that means in the long run, in the long time, they will probably have the side effect on the other organs. Slide, please. And also because we know that the function in the brain is very complex, so the depression situation will give also the stimulation in the other places. Next, please. For this, for example, next slide. If the threat goes, we know this is the fight or flight situation. But if it's not succeeded, we'll have the freezing. So we have the tonic immobility. So this is also known in the rape situation that will happen like this, but people will give the different idea about the tonic immobility in here. Slide, please. There is a studies done in 2018 in the up panel shows that they got the harassment in less than 5 years old, and now in the studies they see is in 51 years old, and below is 31 years old. In this study, actually, even they give the psychotherapy and the hyperbaric oxygen treatment for 6 months, and every 2 months they have to draw about the situation in their body. And we can see here there is always shows something with the genitalia organ there, system organ, and it seems that the panel, the up panel says that they never smile. And the similar situation also can be seen down the panel here. So it means that the post-traumatic stress disorder can occur the long of their life. So that situation, next slide please, will give the impact and can be happen in early life stress, and we usually call the allostatic process. That means the interaction of the situation will give you the imbalanced organism, so the homeostasis can be failure, and then can develop some diseases. Next slide please. So we know that this in the neurobiology theory of depression can have the 6 pathway here, from monoamine, from dopamine, neutroprin, inflammation, gut microbiota, interaction of hypothalamic pituitary adrenal, and inflammation, also the glutamate. So from these pathway, actually we can develop something that can cut the circumstances in a negative pathway, so that will not happen the depression, because the depression itself can make the victims will not be able to speak or to tell anybody what actually happened to them, and then they will take times until they have enjoy or the enjoyment of the body, and they develop more depression. So the depression itself will continue or will decrease. Next slide, please. And we have the dopaminergic pathway. This is well known. And if people have some medicine, usually the side effect of the medicine itself will influence the situation, like feeling very weakness and sleepy. So the pathways through the necrostriatal, mesolimbic, mexacortical, tuberocondybular, that is have the function on dominant motoric, and then memory and learning, inhibition. So this is dopaminergic pathway. Actually, is the best known pathway in the depression. Next slide, please. And if we can see down to this situation that could, through the sympathetic activity, the depression can happen through the sympathetic activity, maternal cortisol, or the NMT. And this we follow the molecular level down here. So actually, if we can also have some substances that can change the number of this molecular level, that will help the people to decrease the depression. This is actually next slide, please. Some studies has been done by Dr. Nia Kurnianingsih to get her PhD now, finished by that, but some there is just some example how she tried to extract the purple sweet potatoes, and then from that is a gift to the maternal stress to have to evaluate whether the pregnancy or the baby of this mother also have a problem with that. Next slide, please. And so this is you can also find out the full of the paper. Next, please. Yeah, so this is some hypothetical theory that's been approved, but it's now still in the processing. And so this is just the how the study is conducted. I'm afraid that I'll pass my time. Yeah. Okay. Next. Okay. Next, please. Yeah, this is just the how is the if the pregnancy right, this is the model of the depression, so that will increase the pregnancy and also decrease the number of the baby, and also the the mortality of the baby. Slide, please. I think this is also something that can be seen, that is the give the anthocyanin as the extract from the sweet purple potatoes that will influence the the situation with this immobilization. Next, please. Yeah, so this is the some theory that can be developed from the how is the mechanism of the depression. Then we can also cut. Next slide, please. In the gut-brain axis, for example, and also index in the presynaptic neuron. And next, please. And so with this situation that can help the people, the victim with depression, not to have the depression by using this kind of natural resources. So in my last take-home message here is, please stop gender violence through the community activation. Next slide, please. And help the victims to, next, yeah. And then help the victims to overcome the depression while preparing to the whole process of the holistic recovery. So usually have to go to the process of justice, thing like that. And we have to trust the victims actually. And not only that, but we have also give something to decrease their depression. So by doing more biomolecular research using specific natural resources to cut the pathway of circular fatigosis of depression, we will have the gender-based violence. Thank you, and this is our team. Good morning, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Okay, thank you very much, Dr. Reti, for a very insightful presentation. I cannot agree more on how we have to help the victims and how we have to help to stop the violence against the women, especially and children as well. Okay, thank you very much. For the audience who wants to discuss more with Dr. Reti about gender-based violence, please hold on, as we are going to hear the second presentation from Dr. Deka Sarmwitayawong. Sorry, I keep on pronounce your name wrong. Okay, but beforehand, please allow me to read the brief biography of our next speaker in this session. Dr. Deka Sarmwitayawong is from Division of Health and Applied Sciences, Faculty of Science, Prince of Songkla University. And he got his PhD in biochemistry and molecular biology from the Pennsylvania State University, whilst his Bachelor of Arts in molecular, cellular, and developmental biology is from the University of Colorado. He has many publications in reputable journals. You can see them in the screen, as well as awards, scholarships, and honors from the Pennsylvania State University and from his country as well, which is Thailand. Okay, and in this session, Dr. Deka is going to deliver his presentation entitled Fungal-like particles inhibit adipogenesis in 3T3L1 adipocytes. Okay, may I ask you all give your full attention to Dr. Deka Sarmwitayawong. Please, Dr. Deka, the screen is yours.
Okay, so I have the video presentation, but it's gonna take like 30 minutes. I can do the live presentation, so that way I can like shut down some of the presentation. Do you want me to do that?
Yes. You can go to share screen.
Okay, give me a minute.
Okay. We can see your slide already.
Actually, wait a minute. So this is the video, but the video will last like 30 minutes. So I can do the live presentation.
You have 20 minutes, and after that we will have 10 minutes question and answer session.
Okay, so I'll do the live presentation. Okay, I don't know if you can see my slide and the mouse that I'm pointing. Can you see it?
Yes, you can see.
Okay. All right. So before I start, I'd like to thank Professor C. Rahayu Lestari and the organizing committee for giving me this opportunity to become a keynote speaker for this meeting. Okay, so the work that I will be presenting today is mainly from this paper that we made it to scientific reports last year. And all of this work mainly was done by my former graduate student, Dr. Chinavi Chakawan Pitak, who is now doing his postdoc at the University of Washington at Seattle. Okay, so the first question is, what on earth is fungal-like particles? So basically, these fungal-like particles are something that mimics the fungal particles. The actual fungal particles look like this. If you zoom in the structure of the cell wall, and you're going to see many compounds on the cell wall. But the most important thing on the cell wall that initiate the immune response is the beta glucan on the cell wall. But this is not.
I'm sorry, Dr. Deka.
Yes.
Forgive me for interrupting, but the slide that you can see is red slide only. I think probably you can start over to present.
Okay. I am sorry, but I thought it came out okay. Hold on. Okay, so
Just
with this
slideshow.
slide, right?
Yeah, I think that's better now.
Okay, maybe I'm gonna need to do it from here because I have no idea, because on my screen it shows one thing. Okay.
Okay.
So okay, I'll do it from here. Right. So where was I? Okay, I was here, right? So the fungal-like particles, and I was talking about beta glucan on the cell wall. So previous publication suggested that the beta glucan on the cell walls are something which is important for initiating the immune response in immune cells. Okay, I'm sorry. Give me a minute. Okay, so many technical problems. Okay, so the idea is if we can create something that mimics the structure of the fungal particles, then we might use them to initiate the immune response. And my previous results, my previous findings suggested that if we use the actual fungal particles, the heat Q yeast, to test the response on the cells, it's doing okay. It's doing quite well actually with the immune cells like the raw cells. But when it comes down to the adipocyte, in which there was no report, previous report that suggests that these kind of cells can respond to the fungal particles. So we tested it and we found that the actual fungal particles can significantly initiate trigger the immune response on the adipocytes, but the level of response is so low. And this is why we needed to create something that mimics the actual fungal cells. So we created the fungal-like particles. And the fungal-like particles came from the conjugation of the short beta-glucans, which is laminarin, and the polystyrene beads. So in the end, we get a sphere with beta-glucan attached onto it. And we call these fungal-like particles laminarin-coated beads. Okay, and this is our previous publication before this paper I presented to you. So we found that the LCB, or the laminarin-coated beads, would somehow bind to the adipocyte and trigger the NF-kappa B pathway, which is essential for the inflammatory response in the cells. And we show that this, the binding or the recognition of LCB by the adipocyte or pre-adipocyte would trigger the phosphorylation of I-kappa B alpha and send it to destruction by the proteasome complex, so that the NF-kappa B complex can initiate the expression of inflammatory cytokines. So with these results, we still did not know whether this LCB can have any effects on the adipogenesis of the cells. And so this is the main question I wanted to address. If the LCB stimulates an inflammatory response in the cells, then does it inhibit adipogenesis? If so, how would that happen? So before I start the results, let's talk about adipogenesis, okay? But before we get to adipogenesis, you need to understand the concept of energy balance. So all of us have to eat and do exercise. If we can maintain these two activities, our weight will be constant as time goes by. But if you tend to eat more than do exercise, then your weight will eventually increase and you start to have spare tire. Okay, and basically the excess nutrients that you eat from food will be stored in the fat cell. The fat cells can be found in what we call adipose tissue. And the biology of adipose tissue is that there are two ways that the fat cells can be stored. There are two ways that the fat can be stored in the fat cells. The first way on the left is for the fat cells to multiply themselves, and basically they will undergo so much adipogenesis to increase its own number. Okay, so in the end you get high sensitivity of insulin, and this is considered good. And basically this process is called hyperplasia, which happens with the subcutaneous fat, so underneath your skin. In contrast, if the fat from the food is stored in the adipocyte, in which the adipocyte did not multiply themselves, but it tend to expand the size, and this is called hypertrophy. And this is considered bad because it will lower the insulin sensitivity and leading to many diseases like insulin resistance and diabetes. Okay, so what is adipogenesis? As I mentioned, adipogenesis is a complex process. This can happen to many cells that store fat in it. So like muscle cells can undergo adipogenesis as well, the fat cells, okay? But I'll just focus on the adipocytes, okay, pre-adipocytes. There are 3 stages of adipogenesis: the early stage, intermediate, and the late stage of adipogenesis, okay? And each of these stages is controlled by different transcriptional factors. For example, on the early stage, in this stage, the cells will undergo what we call the mitotic kernel expansion. So basically, the cells will undergo almost 2 rounds of cell divisions to multiply themselves. And there are 2 major factors in this early stage, which are the CEBP beta here and the CEBP delta. And these 2 factors are important for the expression of the markers of the intermediate stage, which are the PPAR gamma and the CEBP alpha. Okay, and these 2 factors, of course, are important for the expression of enzymes responsible for fat storage, like the FAST, the fatty acid synthase, or the LPL. Okay, so those are the adipogenesis. Okay, and we began our investigation basically by having the differentiating pre-adipocytes, and we introduced the LCB, okay, to the system and asked the question whether this LCB can inhibit adipogenesis after the process of adipogenesis has completed. So in this picture, there are so many panels. Panel A's are just the pre-adipocytes. This is without differentiation. So after differentiation, we stain them with the dye called oil red ore, and this is gonna give the red color because it can stain the fat in the cells. Pre-adipocytes doesn't have any fat, so it doesn't get stained well. Panel B is the fully differentiated cells. So you get a whole bunch of red color on the cells because they are fat accumulated. Panel C's are the negative control, so we use the uncoated beads, basically just a blank bead without beta-glucan attached onto it. So differentiation takes place without any problem. Panel D right here is the positive control using the LPS because we already know that LPS inhibits adipogenesis. So from E, G, and F, E, F, G, okay, these are the experimental set in which we titrated the amount of LCBs, and as you can see, as we increase the LCB, there will be less red color stain to the cells, and we also dissolve this red dye with the DMSO and do the measurement using the absorbance, which show on panel H, and you can see that as the LCB increased, the amount of the red color started to significantly decrease. Panel I are the quantitative measurement. We use the TG assay. We measure the amount of triglyceride assay in the cell. So these results correlate with the oil red ore staining very well. Okay, so from this experiment, we show that LCB inhibits adipogenesis. And the next question, oops, sorry. Okay, it doesn't show up because okay, anyway, so I'll do it manually. Okay, so the next question is, we try to ask, could this inhibition happens at any stages during adipogenesis? So the idea is we have this timeline, so the 8 days of differentiation. So we introduce LCB at different time periods. So A is right from the beginning, B is from day 2, C is from day 4, and D is from day 6. And we did similar experiment. We did the orbit or spinning on panel B, and the result shows that only if we introduce LCB to the early stage, then we can get the efficient inhibition of adipogenesis. But if we introduce LCB at the later time point, this inhibition is not effective. And we also perform the gene expression by looking at the expression of PPAR gamma and CEBP alpha. And the results also agree with the Orbit staining results, in which only the situation in the A, in which the LCB was introduced at the earliest stage, then we get the down regulation of these transcription factors. Okay, so the results suggested LCB inhibits adipogenesis in the early stage of differentiation. Okay, bear with me because all this animation wouldn't work with this regular mode. So next we analyze the protein expression in the differentiating cells. Okay, we perform the time-dependent effects and we perform the dose-dependent effects of the LCBs, and the results will agree with each other that the presence of LCB inhibit the protein expression of these guys. So CEBP beta, CEBP alpha, PPAR gamma, FAST, FABP4, and ACC. Okay, so we see the effect on the time-dependent, different timing, and we also the effect as the dose. As we increase the dose, the inhibition is more pronounced. Okay, so this experiment lead to the conclusion that LCB inhibits the expression of key adipogenic transcription factors and enzymes, and in the differentiating adipocytes in dose and time dependent manners. Okay, so this is a bit complicated slide because it basically shows the flow cytometry results. We asked the question whether this LCB can affect the mitotic clonal expansion. So the first experiment, we did the flow cytometry analysis. So for those of you who are not familiar with this kind of experiment, basically we stain the cells with propylium iodine, which binds it to the DNA, and then we run the cells into the flow cytometer, and the machine will detect how much DNA is present in the cells. So normal cells on A, this is a pre-adipocyte. You see a lot of cells are in the G0 or G1. So this first peak on the left is very high, and only a small population of the cells are in the S phase. Okay, so this area is pretty low, and some of the cells are in the G2M, in which the cells already duplicate the DNA amount, so it's got the twice as much the DNA content. Okay, so A is the regular cell pre-adipocytes. Okay, so the B is the negative control, is the untreated. So we differentiated the cells. Okay, and the cell started to enter the S phase. So the cell started to replicate the DNA. So you get more area under the S area, right? And C is a negative control too, because we use the unconjugated beads. Okay, so the results came out very similar to the untreated. So this is saying that the unconjugated beads has no effect whatsoever. Okay, and then we began to titrate the amount of LCB. So I want you to compare DEF to A. Okay, so you can see that as the amount of LCB increases, we get less and less cells population that enters the S phase. So this is suggesting that the mitotic clonal expansion was inhibited. And if this process was inhibited, then we would expect the less amount of the cells after differentiation too. So this result is shown in panel I on the right-hand side. So without any treatment, the untreated cells or the with the unconjugated beats. The graph superimposed with each other, and as we increase the amount of LCB, then we get less and less amount of cells that were present in the plate, in the well. Okay, so suggesting that the cell number were inhibited because the mitotic clonal expansion was inhibited. Panel J was to stain the DNA with a kit, and we quantify how much double-stranded DNA were present, and the result was consistent. As we increase the LCB, we get less and less DNA present in the cells. So all this data suggests that LCB blocks cell cycle progression and inhibit proliferation of the differentiating cells. Okay, now I have difficulty moving the slide. Okay, before I show the next slide, let me give you some more backgrounds. Okay, so the LCB has beta-glucan, and beta-glucan can be recognized by a number of receptors on the cell surface. So much of this knowledge came from a study in the context of the immune cells. Okay, but the knowledge about how adipocytes recognize beta-glucan is very limited. Not so many people are looking at this, and we still don't know for definite what exactly is the receptor for the beta-glucan on the adipocytes. Okay, but there are two. I want you to get your attention on the two receptor. So the first system of the receptor is the TLR2, okay, and the second one is the dectin one. TLR2, like many other TLR or toll-like receptor, require the adapter called IRAK, which is not present in this picture. Sorry about that. Okay, and the dectin one required the adapter called SYK, which is the kinase. Okay, the idea is if we can use specific inhibitors that disrupt or that interferes with the function of one of these or both adapter proteins, then we would get an idea of how the signaling cascade is working in the adipocyte. Okay, so as I said, we use specific inhibitors, and so we chose two inhibitors in this system. One is the IRAK1/4 inhibitor, which inhibit IRAK proteins, which is a kinase, and the other one is the SYK inhibitor. Okay, and the third inhibitor is basically the positive control from the previous study that we had, the IKK2 inhibitor, which inhibit the phosphorylation on the I-copper-B alpha, so thereby stopping the activation of NF-copper-B pathways. Okay, so it should block the inhibition of adipogenesis as well. Okay, so results on panel A, basically we have the differentiating cells set up, and we added LCB at the earliest stage of adipogenesis, and then we include the inhibitors from IRAK1/4 inhibitor, SYK inhibitor, and we perform the IRAK staining to look at how much lipid was accumulated in the cells. And without the inhibitors on this bar, the white bar, you see the lipid accumulation was inhibited, right, in the presence of LCB. However, with the presence of inhibitor and the LCB, you get more lipid accumulation. So you compare this white bar with this striped bar. Okay, so in all cases, you see significant increase of the lipid accumulation. So this result suggests that the IRAK and SYK pathways mediate the signaling cascade stimulated by the LCB. On panel H, so I didn't show you so many results because of the time constraint. Okay, so on panel H, instead of looking at the IRAK staining, we look at the protein expression, we look at PPAR gamma, we look at CEBP alpha. Okay, so in the presence of the LCB, the expression of these two proteins were inhibited, but in the co-presence of the LCB and the inhibitors, you start to see more proteins appear in the Western blot. So this data on the Western blot was translated to that chart, okay, and we can see the effect more clear this way. So, okay, so conclusion is this IRAC and SYK pathways are playing a role in the signaling cascade. Okay, so the next result is to ask, what is the role of AMPK? And you might ask, what on earth is AMPK? So AMPK stands for AMP activated protein kinase. This protein is a complex, okay? And it will function whenever the cells has some kind of activity that use the ATP. So as a result, the amount of AMP increase and the amount of AMP decrease. So with the elevation of AMP, this complex will be activated. So this complex is essential for many kinds of activities in the cells, including the DNA repair, autophagy, cancer progression, okay? But let's take a look at this. AMPK has so many targets, and one of the targets is the ACC proteins, okay? So ACC protein was shown earlier in this presentation. ACC is playing a role in the fatty acid synthesis. And it turns out that in this slide, AMPK inhibits ACC1 and ACC2. Therefore, if this AMPK inhibits ACC, so with the activation of AMPK, then you should get less lipid accumulation in the cells. So with this knowledge, we wonder if AMPK would play a negative role in the adipogenesis. Okay, so we perform the experiment by introducing the LCB. Yes.
I'm sorry for interrupting. Just to remind you that the time is limited here. So could you please?
Okay. I'll hurry. Okay. So okay. Yeah. So on the Western blot, in the presence of LCB, you can see more AMPK phosphorylation increase as the time goes by. Okay, and you see more ACC1 phosphorylation as well. So it suggests that AMPK plays a negative role in adipogenesis. Okay, and we also use the inhibitors, and in the presence of inhibitors, you see less phosphorylated AMPK appears in the system. Okay, so this result suggests the role of AMPK in the adipogenesis. And the last results I wanted to show you is the beta-glucan receptor. The previous publication, actually this is the short communication, suggested that CD36 may be a receptor of the beta-glucan on the adipocyte. And so we asked question whether this CD36 is actually the receptor. So we performed the experiment in which the idea is if such receptor is important for the recognition of beta-glucan in the cells, then by introducing LCB, that gene expression of the receptor should be changed to. So we look at the qPCR on TLR2 gene, Dectin1, CD36, and TLR4, which is a negative control. And we found that only the expression of TLR2 and Dectin1 were affected by the presence of LCB. CD36, in contrast, which was previously suggested to be the potential receptor of the beta-glucan on the adipocyte, did not change the expression patterns. Okay, so this data suggested that TLR2 and Dectin1, but not CD36, are potential candidates for beta-glucan receptor in differentiating adipocytes. Okay, and this is basically a summary of the mechanism that we propose. I'm not going to go through the detail because it's going to take so much time. And this is toward the end of the presentation. I'd like to thank our collaborator in Japan, in Okinawa, Professor Hirosuke Oku and Dr. Inafuku, for helping us with some of the experiment. And I also would like to thank the Department of Biochemistry at PSU, the Prince of Songkla University grant, the Science Achievement Scholarship of Thailand, SAST, the PhD Overseas Thesis Research Scholarship, Prince of Songkla University, and my current lab members. Okay, thank you very much for your attention. I'll be happy to take any questions that you may have from the presentation. Thank you. Sorry for going over.
Okay, applause for Dr. Jeka Sarmutayawo. Okay, thank you very much, Dr. Jeka, for giving such informative and interesting presentation. Okay, ladies and gentlemen, we are coming to the very short question and answer session. So I'd like to invite probably one or a maximum of 2 questions. Okay, there's a question from the participant in the slides. Okay. The participants at corner and at the back. Okay, please mention your name and to whom the question is addressed to.
Assalamu alaikum warahmatullahi wabarakatuh. Actually, my name is Hussein Muhammad Taher. I'm from Sudan. I study university biology, master degree. So my question for my supervisor or my doctor, I agree with her. We have to stop the violence against women. But the question here, can you like explain to us why there is violence against women? For example, in the Middle, in the East, we know gender equality, gender equal. So especially for teenager. So sometimes, as I see, there is a revolution or demo. So we have to stop the violence against women. I agree with that. But in the same time, in the East, they say women must be equal to men in everything. So in Islam, we know women have a limit. So if we agree with this, with what the East they say about that, so that means we give the woman the freedom to do what she can to do. So for her, she can do what can do, especially teenager. You know that the teenager is the how to say, like this is a target, because teenager at that age they can do everything. So they said they don't use niqab, wearing short clothes and so on. So my question here, like, what makes this violence? For my opinion, sometimes gender equal. This is one of makes the violence against women. So yeah, can you explain to us? Thank you.
Okay, Dr. Reddy, could you answer or give response to the question?
Okay, thank you. I'm very sorry that I cannot hear you properly, but maybe if I'm wrong, please give me some clue. But in the last time that you said that they why or gender-based violence can be stopped by the gender equivalence or gender equal. This is your statement lately, sir?
What do you say?
Yeah, you said that gender-based violence can be stopped by the gender equal, right?
I mean, I agree, we stop the violence against women. But
Yeah.
at the same time, if we look right now, most of the women, they need the gender will become equal, the East. Because in the East, they give
Probably straight to the question. Straight to the question.
my question, what the make this violence against the gender, against the woman? So this is my question. Why is the violence against woman? And also, I
Okay.
have like, yeah.
Why? The answer is only one actually, because in equal situation that makes the gender unequal, that will make the gender-based violence happen. Why? Because the equivalent here is not only the situation that okay, men and women can work in the similar professional and similar whatsoever, but one thing that the mindset of the community has not changed yet. It seems that the woman subordinate to the man, so that's why the difference of this power that will give the violence against women. That is why.
Okay, I agree with you. But I mean also, if we give them the chance, even in Islam say woman is not like women. Here they take this by different way. So they say we are same in the everything. Every duty man can do it. I know it, woman can do it. So in the East, woman we cannot like say to woman don't do this and don't do this. But in Islam we have a rule. Woman must be woman. So if Rasulullah SAW says that woman is not become equal with man, this is one of my this is one I want to say.
Yeah. We got the point. Yeah, I think it will be a very long discussion discussing about the gender equality and stuff like that. Yeah, but I'm so sorry we are limited with the time. Okay, I'm so sorry. And I believe that Dr. Eti is open for discussion with her offline. Yeah, so probably you can contact her directly. And also there's a question in the chat box. I'm sorry, I think the time is finished here. So to Pak Ibnu from Brawijaya University, I hope Dr. Eti would like to answer the question in the chat box in person. Yeah. I think that's all, ladies and gentlemen. We finally come to the end of the second plenary session. But before we close the session, I'd like to thank you very much to Dr. Eti Ratnawati and Dr. Jeka Sermita Yawong, our keynote speakers in the session, for sharing their fascinating research and knowledge with us today. Thank you very much. And thank you all participants for attending this session, and have a great conference. Okay, wassalamualaikum warahmatullahi wabarakatuh.
Thank you for Ms. Nani Farida. And well, that was our second plenary season from Dr. Eti Ratnawati, an Indonesian Psychology Society and National Commission on Violent Against Women, and Dr. Deka Sermiwita Yawong from Prince of Songkla University. Thank you for your amazing knowledge you've shared.
All right, ladies and gentlemen, now we step to the last plenary session, which is the third plenary session. In this session, we have Professor Dr. Chanpen Chanchao from Chulalongkorn University, and also Professor Dr. Rob H. Marsh from University of Liverpool as the keynote speakers, and also Dr. Vivi Novianti, MSI, as the moderator in the session. For Dr. Vivi Novianti, the time is yours.
Okay, thank you very much. So welcome to the third plenary sessions. Welcome, ladies and gentlemen, and this is International Conference on Life Sciences and Technology 2022. My name is Vivi Novianti, and I'm going to be the moderator for this session. So it's been more than 2 years, right? We're facing the pandemic situations, and 2 years earlier, ICALIS always conducted online. So finally, these years we do by hybrid, and hopefully that this conference can give us new insight and have a contribution for the pandemic situation and also for the science development. And State University of Malang, one of the big universities in Indonesia, wants to give a positive contribution to the situation nowadays that we're facing. We have 2 speakers who are with us. The first one, we have Professor Dr. Chanpen Chanchao from Chulalongkorn University, Thailand, and also Professor Dr. Robert Hunter Marsh, as known as Professor Rob Marsh, University of Liverpool. Will be joining us later on maybe. Okay. And first, I'd like to greet Professor Chanpen Chanchao first. Hello, Professor. How are you? Okay. I hope I don't mispronounce your name. Is it correctly? Okay. Thank you. Okay. Ladies and gentlemen, we will see the presentation till the next 40 minutes. And all the keynote speaker will talk around 20 minutes. And after the keynote speaker have presented their topic, we will have discussion for about 20 minutes. And for your information, our audience, our faculties, students, researchers, and also NGOs, which is quite various background. And for the audience, if you have any questions, you can ask your question during the question answer sessions by raising your hand, or you can type in the chat box if you join in the Zoom link. Okay, I will read them for you. Okay, and I will read the profile of Professor Chanpen. Okay, it has many achievements here. I just read some. He is from Ayutthaya, Thailand. And he got her bachelor degree in biology from Chulalongkorn University, Thailand, and got her master and PhD in biology in the field of molecular and cellular biology from the same university, Virginia Polytechnic Institute and State University, Virginia, USA. And okay, and she has many awards. I just read some. National Research Award, Good in Level Agriculture and Biology in 2022 from National Research Council of Thailand. And then this one that I want to highlight, Young Outstanding Researchers in Biological Science, Faculty of Science, Chulalongkorn University in 2008. And also she has a research fellow, many research fellow, and also a lot of publication that I cannot mention here. Okay. Okay, That's all about her. Okay, Professor Chanpen, you have 20 minutes to present your work, so please.
Morning, everyone. It's my pleasure to have a chance to give you a talk today. The title of my talk is about bee products in Thailand and their bioactivities. Okay. I think the slide is not on the big screen yet. Excuse me. Okay. Yeah, sorry for the problem. Okay, so it's my pleasure to have a chance to give you the talk today. The title of my talk is Bee Products and their Bioactivities. Okay, the next one, please. The next slide. Okay. So about the work in my lab, actually there are 3 scopes in the lab, but today I will talk only 1 scope. So it's about bioactivities of bee products. The next one, please. Okay, as you know that for decades, I think we moved to the aging society, right? So for example, look like Japan is a hyper-age society already. But look about Thailand, right? So in the next 20 years, the amount of aged people will turn to be double. So from 13% to 26%, okay? So what should we do about it? I think it doesn't matter how long we live, right? But it does matter how healthy we will be when we are old, okay? The next one, please. Okay, so when we look at the disease happen to us, you know, especially for old people, it's involved with many enzymes. And if we go into the detail of the drugs, they are enzyme inhibitors, okay? The next one, please. Okay, so for example, for type 2 diabetes, right? So it caused by 2 enzymes. The first one is the function of alpha amylase, and the second one is alpha glucosidase, right? So if they function abnormally, it causes the high content of glucose in our blood, right? And it causes a lot of symptoms to us. So when we look at the drug that used to treat diabetes patients, recently the most efficient one is acarbose, right? And look into the detail how it works for this drug. It's alpha amylase inhibitor, okay? So although it's very efficient in the market nowadays, but it still causes side effects to the patient, right? So it can cause itching or skin rash, stomach pain or diarrhea kind of. The next slide, please. Okay, so go to the obesity one. The next one, please. Okay, so for obesity, so look right now, so that we have plenty of food, right? So for this conference, I ate a lot of yummy food. So I enjoy eating it. You know, so let's bring to the abnormal or excessive fat accumulation in our body, right? So we can gain weight easily, and we have a problem of obesity. A lot of people get fatter and fatter when they are older and older, okay? So one of important enzymes that play a critical role for the fat accumulation in our body is lipase. And if you go to the drug that used to treat that patient, it's orlistat, right? So looking into the detail of the drug is a lipase inhibitor, okay? Also, it's an efficient one, but it still cause side effects to the patient, right? So it can cause flatulence, abdominal cramps, or fecal urgency like that. Okay, so the next one, please. And the next one. Okay, so since we don't have much time, so I can skip a lot of slide, I think. The next one, please. The next one. Okay, so since a lot of drugs we use nowadays cause the patient side effects, right? So that's why searching new or alternative compounds is still important to us, okay? So besides side effects that we get when we take those kind of drugs, you know, nowadays, I don't know whether it's happened here or not, but in Thailand, we import a lot of drugs from abroad. So that's why the price of those drugs are very expensive, right? So it's really hard for some people to buy those drugs, okay? So we should help them, right? Okay, so if we can find some new or alternative drugs that we can make it by ourselves in our own country, that will be good, right? So we can help them to access to the drugs with maybe the lower price or with the safer effects, right? For the safety, the more safety to the user. Okay, the next one, please. Okay, so in our lab, we have focused on bioactivities of bee products. So I think everyone are used to bee products, right? So you are used to with bee pollen, right? Wax, bee venom, royal jelly, honey, yeah, honey, everyone is used to this one and propolis. Recently, in my lab, we have focused on propolis and bee pollen a lot, okay? The next one, please. Okay, so for natural products, including bee products, is practical and benefit in both crude extract and pure compound. So it depends on what choice you are looking for, okay? So for crude extract, it's good in terms of synergistic effect, because if it works well, so that means in that sample, it needs a lot of things to work or support each other, right? And if that extract is work well, normally the price is cheap because you don't have a lot of method to prepare it. And local people can do it easily, right? The way you do the ethanol extract of propolis, for example, everyone can do it if you know how to do it, right? And that's cheap, right? So for honey, if you know that honey has an antimicrobial activity, so whenever you have the wound and you live far away from a hospital, you can use honey at the preliminary use to protect the bacteria infection, kind of, okay? But how about pure compound? So it's good in terms that compounds were already removed, right? And you know the exact compound with the known chemical structure, right? So you know how to control the quality of it very well. So you know for pharmaceutical companies, they prefer pure compounds, right? Because they know the exact compound that works for that symptoms, and they can control the quality well. So as long as you know the chemical structure of that compound, you don't have to go back to the raw material anymore, right? You just synthesized that compound directly from the lab, and how to say, you can sell it with the good exact concentration that used for the effective dose, right? The next one, please. Okay, so for today, I will talk about bee pollen only. So it's the work from one of my PhD candidates. So although we have the pandemic for about 1.5 years, the campus has been locked down, right? We just have a chance to go back to the lab the early of this month. But this person can produce 4 publications within about 2 or 3 years. So she's really amazing for me. The next one, please. Okay, so overall about her work, she collects bee pollen from many types of flowers. So at the beginning, we have to make sure that we get the exact flower. That is the main component on the bee pollen, right? Later, we do the crude extract, and then we move to the partition extract. So in the partition extract, we assay the amount of total phenolic compound and flavonoid content, since many articles have been reported that major active compounds in natural products belongs to the group of phenolic compounds and flavonoids. Okay, so at the partition extract, we assay the bioactivities of 2 groups. So the first one will be for enzyme inhibitory activities. Like I told you earlier, that a lot of diseases are involved or caused by the function of enzyme, right? And many drugs are enzyme inhibitors. So in this case, we paid attention to 5 enzymes, like I show you on the slide there. And then we move to the antioxidant activity. So we use 3 methods to answer one question. Okay, so right now, today, especially in Bangkok, we try to move to be the industrialized area. And, you know, we have a lot of cars, so the air pollution is kind of bad. We have the problem of PM 2.5, kind of like that. So many free radicals in the atmosphere, and it's caused a lot of problems for our health, right? So that's why in this case, I focus on it. The most active partition extract will be analyzed by the high performance liquid chromatography, or HPLC, in order to try to get the compound of the active one. Okay, so that's the overall of the work. The next one, please. Okay, so we go to collect bee pollen from many locations in Thailand, right? So, you know, a lot of areas in Thailand turn to be mono-agricultural place. So look like in December, in winter season, in Lopburi province, you can find a lot of sunflower fields. So you just move the hive of bee to that area. And then you put the box, the pollen trap in front of the beehive. So within 30 minutes, you can get a whole bunch of bee pollen of sunflower kind of. Okay, and we go to many places in Thailand. You know, since Thailand is a small country, so you can go anywhere easily. Like from Bangkok, go to Chiang Mai, you can take only 1 hour kind of, or you go to Udon Thani, it takes around 1 hour kind of by plane, or look like maybe 5 up to 6 hours by car kind of. Okay, so we go to collect pollens from 6 follow pollens. Okay, the next one, please. Okay, so we have to do the detail of sample collection because, as you know, that biogeography or geographic distribution of the sample play an important role for active gradients in natural products, right? Different weather, different location, and different extraction methods can be the key roles to get your active compounds, right? So in this case, we have to record the collecting site, the exact geographic location or collecting time like this. The next one, please. Okay, so although we go to get bee pollens from mono agricultural area, we have to do the confirmation whether we get the exact follow type of the pollen, okay? Because as you know that the major gradient in bee pollen is follow or flower pollen, right? Okay, so in this case, we use the scanning electron microscope to look at the follow pollen in bee pollen. So the pictures are very beautiful, right? And very unique. So look like in figure A, you get the follow pollen of T. So we can make sure that the bee pollen from A is majorly or is mainly composed of T follow pollen kind of. Okay, so this one is the way we can confirm the raw material, okay? So either way, you can do the molecular analysis by doing the partial sequence of the ITS gene, okay? Of the ITS of the gene, okay? And in this case, I don't show you that data. I think this one should be enough to go on. Okay, the next one, please. Okay, so in all natural products, you know, including bee products, there are a lot of chemical compounds in it, right? So for example, in propolis, it has been reported there are over 300 identified compounds. And you know, most of them belong to the group of phenolic acid and flavonoids. So that's why we have to focus on this assay as well, okay? And I told you earlier that major active compounds or our interesting bioactivities can depend on external factors like collecting location, extraction process, or internal factors like bee species kind of. So at the same place, okay, you have the same flower fields, but you use different bees to go to collect the bee pollen. You may get different active compounds in the pollens you collect, okay? The next one, please. Okay, so this is the how to say the brief method to do the crude extraction, okay? So we just use universal solvent, which is methanol or ethanol, to do the preliminary extraction, and we get the crude like this, okay? The next one, please. Okay, and this is the way we do the partition extract. We use the separating funnel with 3 organic solvents. 3 of them are different in polarity. For example, hexanes. You know, not that one, I'm sorry. It should be the one before this. Yeah. Okay, sorry for this problem. Okay, so for partition extract, we use the separating funnel with 3 organic solvents. Look like, for example, hexane, as you know, that it contains no polar at all, right? While dichloromethane is a medium in polarity, and methanol is high in polarity, okay? So at the end, you can get 3 partition extract from your sample. The next one, please. Okay, so after we get partition extracts, we have 6 bee pollens, right? We did the partition extract with 3 organic solvents. So total, we have allowed 18 partition extracts, okay? We assay total phenolic compound and flavonoid content. So overall in the table, you can see that we get the highest amount of total phenolic compound and flavonoid content from the solvent of dichloromethane, right? And among 6B pollens, you can tell that we get the good data from mimosa bee pollen, right? So for this case, we take the mimosa bee pollen, continue for the purification. The next one, please. Okay, so part one, we estimate the enzyme inhibitory activity, right? Look like for anti-alpha amylase activity, it will be good for the field of type 2 diabetes like this, okay? The next one, please. Okay, we move to table 3. So you can tell that our 16 partition extracts are not good for anti-alpha amylase and alpha acetylcholine esterase at all, right? But we found that dichloromethane partition extract of mimosa bee pollen is good for anti-lipase activity, right? So we do these things by comparing to the positive control, which are drugs that use in patients. Look like for anti-amylase activity, we use acarbose, right? And for anti-acetylcholine esterase, we use physostigmide. That is the drug in the market that a doctor can prescribe a patient to go to get it. And for anti-lipase activity, we use orlistat, right? So for orlistat, if you are used to it, it's really, really expensive, okay? Okay, so in this case, among 3 enzyme inhibitory activities, we pick up the anti-lipase activity to go on, okay? The next one, please. So how to do the purification? Just use the column chromatography, okay? So you can vary them into the different diameter of the column, the difference in the length of the column, or the difference of the resin in the column, right? You can use the big size of the beads, the smaller size of the beads, kind of, you can change different solvent to elute your sample. You can change the time to collect your fractions, kind of, okay? So this is just the brief detail for the purification by using the column chromatography. The next one, please. Okay, so what we got for anti-lipase activity. So we did determine the half maximal concentration of inhibition or IC50 value, okay? So after we did the first purification by using the silica gel 60 column chromatography, all right, and look at the second time of the purification, you can see that in the column on my right hand, look at the IC50 value. So it seems to be that the more purification, the better in IC50 value we can get, right? Okay, the next one, please. Okay, so how can we determine the purity of the compound we get after we do the purification by using any column chromatography? We use the thin layer chromatography, right? And you just, if you can see, just some bands, not a whole bunch of bands, right? Or only single bands come up on the TLC plate. So that's good. Look like you get pure compounds already, right? For this case. Okay, the next one, please. Okay, so whenever we get the pure compound by tracing them using the TLC plate, we use, in this case, we do the gas chromatography connecting with the frame ionization for detection analysis, okay? We found out that the most active one, the most active fraction after we do the after we did the second purification by using the column chromatography, okay? So we use the GC connect with frame. And we found out that we got a lot of, the fatty acid, unsaturated fatty acid, around 16, okay? So we analyzed more. We found out that among those 16 fatty acids, we can find one saturated fatty acid, which is palmitic acid, and in D, right? And we get two more unsaturated fatty acids, which are linoleic acid and alpha-linoleic acid. So both of them are unsaturated fatty acid, okay? So in overall, we can tell you that in mimosa bee pollen, if you extract it with dichloromethane, you did the purification by maybe second time of column chromatography, and then you did the GC connected with frame, you can get three active compounds of fatty acid that can have the anti-lipase activity. Okay, so move to the part 2. We pay attention to antioxidation, anti-lipoxygenase or LOX activity. So this one involved with asthma disease, right? For lipoxygenase, if it's function a lot, it causes you to have asthma, right? Okay, for anti-tyrosinase activity, it's involved with the hyperpigmentation. So look like we live in the tropical zone. We are overexposed to the sunlight a lot, right? So that's make our skin make a lot of pigments, right? So that's why we have kind of brown to dark skin. So maybe we don't want our skin to be too dark. So we have to find something to reduce the melanin in our skin, okay? All right, so move the next one. Oh, okay, sorry. Okay, I'm sorry. I talked before slide, but okay, anyway, okay, come to this one. I'm afraid that I cannot finish it in time, sorry. Okay, so for antioxidant activity, we have 3 methods to detect the free radicals, okay? So we use the PPH, ABTS. So if you are familiar with ABTS method, you can measure them into 2 ways. So you can compare it with the Tolox, right? Or you can do it into the different way to determine the effective concentration at 50% or EC50, okay? And or you can do the FRAP assay. All of them are colorimetric methods, right? You just see the change in the color and you measure the absorbance of them, okay? We use 3 methods because they can detect the different free radical. But if you look at the data on the screen, you can tell that all of them give us the supportive data, right? So again, you can tell that the dichloromethane partition extract of the mimosa bee pollen give us the best antioxidant activity, right? Okay, the next one, please. Okay, so how about anti-tyrosinase activity and anti-lipoxygenase activity? So for this one, for LOX-I, anti-lipoxygenase activity, the same sample. So seem to be mimosa is very interesting for us, right? You know, previously mimosa was wheat plants in Thailand. But now today, or maybe recently, we changed our mindset. And we know that bees love mimosa flowers a lot. So many bee farmers in Thailand, we grow mimosa for our honeybees. Okay. All right. So if we go to the column of anti-tyrosinase activity, you can find out that dichloromethane partition extract of sunflower bee pollen give us the best activity. Okay. All right. And the next one, please. Okay, so we do the purification. In this case, we use one column chromatography. So you can find out that DCM partition extract of the mimosa in the fraction number 3 give us the best activity in antioxidant, right, and anti-lipoxygenase activity. Okay, so from the previous one, we purify by 2 column chromatography. But for this one, we found out that 1 column is enough. And then we analyze the sample by using the HPLC instead. Okay, so the next one, please. Okay, so what we get from the HPLC. So the chromatogram like this, you can find out that at the retention time around 15.3, right, and 20.1, the peaks are very interesting to analyze the chemical structure, right? Okay, so the next one, please. We found out that we get the spermidine derivative in those fractions, in both fractions, which are very active in antioxidant activity and anti-lipoxygenase activity. Okay, so the next one, please. So remember that besides mimosa bee pollen, sunflower bee pollen is very interesting for anti-tyrosinase activity, right? Okay, the next one, please. Okay, so we do the same thing. We move to the purification. From the first column, you can tell that allow fraction number 4 and number 5, they have good activity for anti-tyrosinase activity, right? The next one, please. Okay, I move so fast because I have around 2 minutes or 1 to 2 minutes left. Okay, so only by single column chromatography, we purify by using the HPLC, right? So this is the chromatogram we got. So we found out that at the fraction 5 after the first column, we found 2 interesting peaks, right? At the retention time of 22.2 and around 23.9, right? Okay, so we use the NMR, the next, please. We use the NMR, the nuclear magnetic resonance, to analyze the chemical structure. And we found out that we get several spermidine A and B in those fractions. The next one, please. Okay, so this one is very interesting because when we compare the IC50 value to the kojic acid, which we use as a positive control, the IC50 values are very interesting, right? So look like for spermidine B can compete the kojic acid very well. Okay, the next one, please. Okay, turn into the conclusion that mimosa bee pollen is good for antioxidant, anti-lipoxygenase, and anti-lipase activity, right? While sunflower bee pollen is good for anti-tyrosinase activity. In overall, you can tell that bee pollen, so previously we always consume them as food, but now we know that it can be alternative source for our interesting bioactivities, right? The next one, please. Okay, so at the end, I would like to thank Malang State University and Mr. Muhammad Andri Priyo Uttomo who invite me, so I have a chance to be here today. The last one, please. Okay, so at the end, I would like to thank you all for your attention to listen to my talk. Thank you.
Okay, thank you very much, Professor Chanpan. It is a very interesting presentation. Propolis, one of the compounds bioactive that contain in bee, honeybee, is contain 300 300 identified compounds only for propolis. So if it's very interesting, we will discuss it later on the question and answer sessions. Okay, thank you very much, Professor Chanpen. Now I want to go to Professor Robert Hunter Mars, or Professor of Mars. How are you, Professor of Mars?
I am very well, thank you. Good afternoon, I believe it is in Indonesia.
Good afternoon. It's early morning at the British time. It's
Yeah,
6
very,
around.
very early morning.
Yes, and it's a fall sessions. Yeah. Okay.
Yeah.
I will read your profile of Professor of Mars. It's very long, 44 pages. And Professor of Mars is a professor of applied plant biology. And okay, so long award or achievements. I just read some of them. Okay, in 1993, he is a president, agriculture and forestry section in British Association, and he already wrote a paper for, I noticed, more than 400 paper in journal, reputable journal, and also read more than 100 for the research report. So I believe and I know him well. So he is no doubt is one of expert in his field. Okay, I think it's time for you, Professor Mars, to deliver your words. Your time is 20 minutes. So.
Right. Okay. New share. Can you see the screen?
Yes, we see it clearly.
Okay, good. Okay, well, I'm going to talk. Can you hear me okay? Can you hear me, Vivi?
Yes, I can. We can hear you.
Okay, good. Well, I'm going to talk today about controlling invasive species, and specifically using an experimental approach. And that's the thing I'd like you to get out of this talk, is the fact that it is experimental. And I'm going to talk about one of the species that I've worked on a lot, bracken. That's the word I'm going to use. It's the English word for the fern Pteridium aquilinum. It covers the whole world, and it originally, a lady called Tryon came up with 2 subspecies and a whole load of varieties. Now, this man here, John Thompson, did a fantastic molecular study of all of these bracken types and came up with a revision. And basically, there are only 4 species, basically a southern hemisphere one, or ones, and the main one is the one I'm going to talk about, Pteridium aquilinum, and the subspecies I'm going to look at is Aquilinum aquilinum. The one in Southeast Asia, which is where you are, is Whiteyanum. Anyway, bracken is a native species in the UK, but it causes a huge amount of trouble because it is so invasive. And back in the early 1990s, my colleague and I, Robin Pakeman, did a massive survey of all aspects of bracken, the bracken habitat. We looked at all the benefits and the pros and all the disbenefits. There were a few positives: landscape, a few species of birds, the stonechat, and some butterflies, a few in a few places. But on the downside, the cons, the disadvantages, there were massive problems for conservation, agriculture. We put a cost on it, £9 million in some areas of England and Wales, forestry, sporting, and specifically animal health, and possibly even human health. And it is generally accepted that bracken is increasing. So, One of the issues is it has at least one, referring back to the previous talk, a very, very potent compound, tequilacide, which has problems with, causes problems for carcinomas and so on. Now, from animal health, there are a whole host of diseases caused by eating too much bracken. Horses, you've got the staggers, rhizome poisoning, you've got cattle, you've got internal bleeding, sheep go blind, and so on. There's a whole host of animal diseases caused by eating bracken. There's also some links to human health, links to human cancers, where it is eaten. There is a high incidence of alimentary tract cancers. People usually eat the crosiers, that's these things here. It's like a bishop's in Western religion, the bishops carry a crook, and that's called a crosier. And you can buy it in many areas of the world to eat, but I would suggest it's not a great thing to try. The habitat also is home to these things, these ticks, and the ticks carry this disease called Lyme's disease, and it is a spirochete called Borrelia burgdorferi, and this is the sort of thing you get. This is my leg. You get a bite in the middle, and you get this bullseye rash spreading out from the edge. I didn't have Lyme's disease, but that's very typical of the symptoms. And one of the questions I'm often asked, is bracken increasing in the UK? And the answer is, I don't know. What I can tell you is there's quite a lot of it. And that's the real issue. And this was the first time that we ever managed to get a decent estimate. In 1990, we did a massive survey, and we showed that there were basically 17,000 odd square kilometers of bracken. And that equates to something like 7% of the country's land surface. So this is although it's a native species, it causes problems over a very large area, and there's another 122,000 kilometers in linear features. That's hedges, roadsides, and along stream banks. So very, very, a very large problem invasive species. So why is bracken a successful plant? And by definition, why is it hard to control? Well, first of all, in the UK, it's a fern. It produces these fronds, and it produces a lot of them. And this produces a dense cover, and this cuts off the light. When these die, it produces a dense litter layer, which smothers everything else. Quite clearly, the fronds tend not to be eaten by animals because it's not good for them, so there's plenty of it. And there's a very large rhizome store underground with lots of carbohydrates, nutrients, and regenerating buds. Bracken in Britain very rarely reproduces sexually. It usually expands by rhizome expansion underground. So it's a very efficient weed species. Lots of top growth, lots of dead material, and a massive store. Now in the UK, the carbohydrates here overwinter. There is no green material during the winter. In the spring, the carbohydrates fuel the frond growth. The fronds then grow to maximum expansion, and then after that, it dies back and transfers the carbohydrates and nutrients back into the store. So to control it, you really need to be able to understand how the rhizomes work. And that's the thing you've got to tackle. You've got to kill off the rhizomes. And you can see here there, I'm much younger there, but the principle is the same. Now, there are basically three types of control method. One is to use mechanical means, like cutting. That's the one I prefer. There is an older technique, a more traditional technique called bruising or rolling, where you roll the bracken and just break the fronds. But cutting I prefer because I think it's a better technique. And you've got some herbicides. There's Ashulam, which is selective. That's the one I prefer. And there's glyphosate, of course, which is non-selective. So if you're in a conservation situation, you want to use Ashulam. Now, and this is really the crux of this talk. The main thing is to test methods of control of invasive species. You need properly designed experiments, you need proper replication, and you need randomization in all the experiments so you don't induce either artificial artifacts or indeed human-induced bias. So very important to have this. So it started off we had a whole host of experiments. Multi-site, we had 2 up in Scotland, a place called Surup. We had 3 at Cannock, just north of Birmingham. We had 1 in Wales, and we had 2 in the Peak District. And this is the experiment I'm going to talk about today. And one of them, this one here, our target was to get a heathland back, and it was set up in 1993. And it was, for those of you who are of a statistical bent, it was a randomized block split plot design. So this is how it was set up. First of all, it was in the Peak District, and we had 3 phases to the experiment, and I'm going to start off with phase 1. First of all, there are 3 replicate blocks, and I'm going to look at just number C. These are the replicates. And within each of those, there were 6 treatments. There was an untreated, that's our control. There was cut once per year, cut twice per year, and they're continuous treatments in that they were applied every single year during this first phase. Then we applied Ashulam once, then cut the next year, then cut once, and Ashulam the next year. So these were only applied at the start. That was in year 1, that was in year 1 and 2, and that was in year 1 and 2 as well. Now, importantly, although I've put this out in order, all of those were randomized within the block. Now, within each of those, there was a split plot design where there's plus and minus sheep grazing. We fenced that area to stop sheep grazing the land. Now, again, those were randomized within each of these. And then within these, we had 3 heather, that's the target species, seeding treatments. One was untreated. We added it as brash, that's just cut stems. And this is where we sucked litter using a big vacuum cleaner from a heathland and put the litter onto the ground. So in total, we had 108 subplots. Now, we analysed this using a thing called principal response curves. And this was my colleague, Joshua Alday, who did this work. And basically, this graph, it's a bit complicated, but it's actually quite easy to understand when you understand what it means. First of all, that's the untreated control. Now, I'm only looking at the main bracken control treatments here, but that's the untreated line. And everything that is above that is positive. And these are the species that contribute to the positive end. These are mainly grass heath species. You can see bracken is way down here. So negative is more bracken, positive is more good species in effect. And you can see that the deviance from the untreated line increases through time. And it gets to this point here, and all of the treatments are significantly better than the untreated line. Now, as it goes on, the cutting treatments retain this significant difference from the untreated control, but the spray treatments come back, and at this point they are no longer significant. So although they're almost the same amount, they have not really decreased much in height. In actual fact, by here, these were no longer significantly different from the untreated control. And this means that only the cutting treatments that were applied every year were causing a significant effect. So this caused us to think again about how we treated the vegetation. We started phase 2. Oh, sorry, there's a summary. The use of principal response curves allowed a rigorous test of alternative state and alternative stable state theory. That's whether you've actually created a new vegetation type or not. But what you need is long-term data from replicated experiments where a treatment effect can be compared to an untreated control. So these are major messages that you do need proper experiments. And with heathlands, we got evidence of an alternative state, but only where you continuously applied cutting. And you had to keep the cutting going every year. So a really difficult and expensive approach. So phase 2. Again, it started in 2004. The untreated was quite clearly untreated. The cut once per year kept going, cut twice per year kept going. Then for these other treatments, which had started to recover, we gave another continuous overspray. We just sprayed the whole vegetation again, and thereafter, every year we spot sprayed every emergent frond. So we went back and looked for individual fronds and sprayed them individually. It was very time consuming, but it was actually quite successful. So in phase 3 was in 2013. We stopped all the treatments in 2012, and we wanted to see how good our treatment had been and how the vegetation would recover after we stopped all treatments. So here's some of the results at the end of phase 2. This is after our first phase of control, where cutting had been moderately successful, had been quite successful. Adulam had been successful, then it had got worse. So we've then applied these new treatments, and what we've found is they've been pretty successful. It's been very good research. Here's the untreated. This is the grazed and the ungrazed. And you can see every single treatment cut once per year, cut twice per year, and all the spraying treatments have been very successful. The spraying treatments have been very, very successful. They're almost zero. The only one that shows a significant difference between grazing and ungrazing and no grazed plots is the cut once per year, where you can see there's a slight increase in the bracken volume. Now, we've used mixed effects models to analyze these data, and indeed all the other data I'm going to talk about during this talk. And mixed effects models takes into account the spatial arrangement of the treatment applications. So that's the, in actual fact, all of these treatments have been very successful. If you look at the effects on the vegetation, now these are not very species rich, it's not a very species rich habitat, but you can see that all the the grazed plots are much higher in terms of species richness, and also in terms of the cover of grasses and other graminoid species, monocotyledons, and also in terms of dicotyledons. So in actual fact, all of the treatments have been quite successful, and they produced a better habitat than the one that was there before. So it's really been very, very successful. And you can see there the effects of the grazing treatment tends to be rather negative on species richness, and also on the cover, or at least for two treatments, on the cover of grasses and graminoids, and one species in terms of dicotyledon, it's slightly lower, it's not very much. So basically, at the end of our phase 2, we had done a good job. All treatments were relatively successful. Well, let's move. So, that's just to reiterate that all treatments have done a very good job in terms of bracken control, in terms of restoration, and the cutting treatments were marginally poorer, especially the cut once per year. So we're getting good information on the best way to handle the control of bracken. So let's stop controlling it and see what happens in phase 3. Well, again, we've used mixed effects models, and this is the effect on bracken, and you can see here that in fact the control is slightly reducing, especially where there's no grazing. And I suspect that that is because of tree invasion, and the trees are having a negative impact on the bracken. But you can see that each of them are starting to recover. That's the blue line where there's grazing. But the one thing that shocked me, and this was actually new information that I didn't predict in advance, is if you remove the grazing, the recovery is much faster. The slopes are much greater, especially in this cut once per year treatment. And that is something that I did not anticipate. And it is important, if you're controlling this species, you do need to have a grazing component to keep the bracken down or to prevent recovery. It also has consequences for ecological theory and how bracken expanded in historic times because of
Professor Roberts.
his time is just about up. Yeah, got it. My timer's just going. So grazing's important. The species number you can see is declining in all of them except the spray ones, and basically in each case the ungrazed is lower than the grazed. So the take-home message: you need manipulative experiments with a control and replication. It's better if they're repeated on multiple sites. You need long-term data, and that is the key. You need funding and you need long-term data. The data analysis is not easy, it's hard, but you need techniques to handle complexity, and you should not shy away from this. In fact, you should embrace it. And thank you to all of these people who have helped me along the way. Thank you.
Okay. Okay, thank you very much for the nice presentation, Professor Roberts. He did for only bracken, bracken for 28 years for doing this research. It's a long-term experimental data he has, so no doubt for he able to write.
I've only talked about my latest experiment. I did start in 1979.
Oh, we have another stuff.
Another
Okay.
stuff, yeah.
So, okay, it's amazing. So, is there any questions from the participant, maybe for Professor Chanpen? Or okay, yes, please. Please mention your name and your department. Yes.
Thank Thank you for the opportunity. My name is Jafar Hilmansyah and I'm from Malang State University. I would like to ask to Professor Mars, how do you make sure every site that you use is in the same condition over the years by one another?
Well, I mean, each of the sites will not be the same. They'll be different. And that's part of why you do need to have different, what you're doing is looking at experiments in different sites, so you are encompassing a range of conditions. Now, what you can do, as long as you've got what's called a Bakke experiment, before, after control and intervention, if you've got before estimates, then you, by definition, you can compare the starting positions of each. Now, we did this with the multivariate analysis, and they were different. Two of the sites were more grass land, and two were much more shrubland. Now, that doesn't matter, provided you know where your starting positions. But actually, this is a fallacy in experimental research, that things should be the same. Because if you are looking at in theory, one should take random positions from the entire population. In reality, you can never do that. You can only put experiments where you're allowed to put them or where you can. But you shouldn't worry too much about the fact that they are not the same, because you're looking at different parts of the distribution of the species.
Okay, thank you, Professor
That's
Mars.
my
I think
view,
it's
anyway.
yeah. So
Okay, thank you,
randomize is very
Professor.
important. Yes. Okay. Another questions from participant? Okay, over there. Please, the committee help to. Okay.
Thank you. I am Randy Francisco Aritonang. I want to ask about bee product. Thank you, ma'am, for the great presentation. Talk about bee is interesting because this creature is very useful for humankind and our environment. So in this case, are we identify the bee first to know they can produce a good honey or a good bee pollen? Or we just identify the honey to know the honey is good? Or the bee can produce a good honey or bee pollen if they have a good environment? Thank you.
Yes, thank you for your questions. Yeah, that's very interesting. So for this case, we just try to find out the advantage of bee products besides consume them as food. Okay, so we are used to bee products as food, look like honey, bee pollen, right? But in this case, we try to find active compounds in it, and we try to show people that those active compounds have interesting bioactivity. So look like what we get from cephalosporin, right? That can present very good anti-telomerase activity. So we may in the future we may do more experiment before we apply it to the cosmetic product or pharmaceutical product kind of. Okay, so that will be a new alternative for cosmetic or pharmaceutical things kind of. So if in the future for some other bee products, if we get some things that we can develop it to be alternative drugs. So we hope that the price will be cheap, that our local people can access it, right? So we don't have to pay a lot of money for imported drugs from abroad. Okay, so that's the purpose of this work. Okay, so the byproducts the other benefits of this work may be if people know how to say the advantage of bee products, people may pay attention to bee farming, right? And maybe good price for bee products, and that support bee farmers for the economic purpose. So I think that's it. Did I answer your question?
Yeah, thank you.
Thank you.
Is there any questions? Again, maybe from the participants, we still have time, 20 minutes for the questions and answer. Any questions from the audience? No? So, okay, one more. Yes, please.
Okay, thank you for the opportunity. Let me introduce myself. My name is Ricky Hikmat. I'm from Biology Department. And in the previous, I would like to ask to the Professor Chang Chao.
Chanpan, yes.
So the professor has mentioned if the bee product has an anti-tyrosinase activity. Would you mind if you explain to me what kind of the anti-tyrosinase activity and what is the relationship between this activity to the advantages of the bee product? Thank you.
Repeat again. What kind of bioactivity?
Anti-tyrosinase.
Anti-tyrosinase. Yeah, and how it relates to bee pollen, right?
Yeah.
Okay. So for this work, we collect 6 types of bee pollens that originate from 6 floral pollens, right? So we assay a lot of enzyme inhibitory activities and almost bioactivities that we focus on were from mimosa bee pollen, except sunflower bee pollen, right? So for sunflower bee pollen, we found out that it performed the best anti-tyrosinase activity, right? So for tyrosinase, you know, it's the key enzyme in the pathway that give us melanin as end product. So for melanin is accumulate in our skin a lot. It causes the dark skin, right? It can cause hyperpigmentation symptoms. So that can lead to a lot of disease. So one of them like cancer or skin cancer. So in this case, we found cephalosporin A and B in sunflower bee pollen. So comparing to the positive control. So in this case, we use kojic acid. Okay. So if you look at the labeling of the suntan lotion, for example, nowadays in the market, you can see the main component is composed of arbutin, kojic acid, like that. Okay. So those are very good, very effective in the sunblock. For the sunblock, it means that it can reduce the activity of the tyrosinase. So that's the key enzyme to produce melanin, right? So if we block that enzyme or delay or decrease the activity of that enzyme, the less amount of melanin in our skin will be. So we will get the lighter skin, or we will get the normal pigmentation, right? Okay, so in the introduction part, I skipped that slide because I'm afraid of the limitation of time. So for arbutin or kojic acid, it's very good for this activity, but it still causes some side effects, like maybe you can get rash or inflammation on your skin if you are allergic to it. So for cephalosporin A and E in sunflower, so right now we know that it has a very good activity comparing to oleic acid. So the next step, we will look at the anti-inflammatory activity. So if it can not cause the inflammation to our skin, that should be the challenging agent to be developed, to be the pharmaceutical things or cosmetic things in order to control the melanin production at our skin. Okay, so that's the purpose of the anti-tyrosinase activity of sunflower bee pollen. Yeah. So I don't know whether I make you more confused or not.
Is it clear?
Thank you. Yeah. Thanks
Okay,
so much.
thank you, Professor Chanpen, for the explanations. I think our time is up, and I would like to make a conclusion from our discussion today that from Professor Chanpen, we can conclude that we get new insight, of course. Bee, is produced honeybee, which is very beneficial for us, useful for our body. And one of the food plants is mimosa, which is, we always pretend them they are weeds or maybe wild, and it is very useful actually. So we have to conserve them to be able to enjoy the honeybee, which is very have many bioactive. And from the Professor Mars, we can conclude that it is very important to have a we have a very comprehensive data. He already proved it for Brecon. He did his research since 1993 until 2021, 28 years only for Brecon, and also he has also many another research. So long-term data is very important. Long-term experimental is very important to prove and also to manage about the invasive species. It's not easy. That's why as experimental studies is very important. So thank you very much, Professor Chanpen, Professor Mas, for joining us. State University of Malang, one of my university, very appreciated, very much appreciated. Thank you very much. And okay, please give big applause for these two amazing speakers. And for you all, the audience, thank you very much to be amazing participant in these sessions, and hopefully to see you again soon, in real soon. Thank you. And after the plenary, we have some souvenir for Professor Chanpen here, and Professor Romars, we will send you the vandal from Universitas Negeri Malang, State University of Malang. We will send it to you to UK. Thank you very much.
Thank you for inviting me.
Okay. Okay, so please, Professor Chanpen, right here. Okay, thank you. So this is the end of our third plenary, and I give it back to the MC. Thank you very much.
Okay, I say thank you for Dr. Vivi Novianti. The award was a last plenary season from Dr. Chepen Chancho from Chulalongkorn University and Professor Dr. Rob H. Mars from University of Liverpool. Thank you for the amazing knowledge you've shared.
All right, ladies and gentlemen, now we step to the next agenda. Here we have a second break. For all the participants, I would like to remind you that break is until 1:45 p.m. And for the online participants, you may not leave this room and you can turn off your camera. Other than that, I would like to inform all participants in this room. During this break, you can have lunch or breakfast. But after that, you wouldn't go back to this hall. You can go to the parallel room, which has been prepared by the committee. You can check it on the abstract book. All right. Setelah ini agendanya adalah istirahat makan siang. Seluruh hadirin dan tamu undangan di sini dapat istirahat untuk sholat dan makan terlebih dahulu. Dan waktunya hingga jam 1:45. Selain itu, setelah istirahat makan siang, para hadirin dan tamu undangan dan juga peserta tidak akan kembali ke ruangan ini karena akan menuju ke ruang paralel yang mana telah disediakan oleh panitia dan bisa dicek pada buku abstrak.
Okay. Then for all the participants withdrawing in the Zoom meeting, the next agenda, which is the parallel session. For this session, we will use the breakout room feature. The operator will split you into a small group and all the participants will be helped by moderator. So we wouldn't go back to main room. Dan kepada partisipan yang ada di dalam Zoom meeting, nantinya agenda selanjutnya adalah parallel session dan akan dimasukkan kepada operator di breakout room. Jadi nantinya tidak akan lagi kembali ke main room.
All right, ladies and gentlemen, we as the master of ceremony would like to say thank you very much for your participation and see you on the second day of ICOLIS 2022. And good luck for your parallel session.
Berdaya saing.
Berkarya yang akan terus berkarya. Nantikan membangun nusa dan bangsa, menjadi rujukan untuk semua. Ayo semuanya ikut bernyanyi. Let's sing together. One, two. Berkarya yang akan terus berkarya. Bangkitkan rindu membangun nusa dan bangsa, menjadi rujukan untuk semua. Excellence in learning innovation. Excellence in learning innovation. Excellence in learning innovation. Excellence in learning innovation.
Welcome to Universitas Negeri Malang, UM. Located in 3 different places, UM provides a comfortable environment for teaching and learning. The main and second campus are located in Malang, surrounded by educational facilities, places for recreation, sports, and business. The third campus is located in the same area as the Shrine of Sukarno, Indonesia's first president in Blitar, East Java. Established on October 18, 1954, UM was born with the name of University of Teacher Education, or PTPG. In 1957, it became the Faculty of Teacher Training and Education, or FKIP. Then, in 1963, it became the Teacher Training and Education Institute, or IKIP. Finally, in 1999, it transformed into Universitas Negeri Malang, which is known to this day. Since its establishment until now, has established itself as one of the 12 educational higher education institutions and is included in the 15 best campuses in Indonesia. One of the visions and missions, is to become a leading university and an authoritative center of excellence in the fields of science, technology, and education. Develops a flexible, adaptable curriculum taught by teachers who guide students and provide skills for the future workforce to become active lifelong learners, independent problem solvers, creators of better communities, and bring education to life. Educates students across 8 faculties. Also conducts special programs such as postgraduate program and teacher professional program. Has been granted an unggulan or excellent by the National Accreditation Agency of Universities or BNPT. Furthermore, there are 4 study programs that have been assessed internationally by AUNQA. And more study programs are accredited by ASIIN and ACUS International Accreditation. As one of the reference universities and a center of learning innovation, continues to develop knowledge and scientific research by providing quality education and training. has also produced many research products that have the right patent, published internationally, and ready to be commercialized. In line with our new tagline, Excellence in Learning Innovation, is the house for top science and technology center in higher education in DLI, or Disruptive Learning Innovation. Disruptive Learning Innovation Center develops innovation in smart learning with research focus on digital transformation on education ecosystem, either in higher education, school, or community, to improve the quality of education service in Indonesia. Many invention and implementation of